Hello? If you're speaking in this session and you haven't signed a consent form or decline form for straightening, could you please come here? Okay.
Okay, good morning. Our first talk is an invited talk by Lisa Giocomo from Stanford University. Please.
Good morning, everyone. Thank you to the organizers for inviting me to speak today. It's a real pleasure to be here. My lab studies our sense of location, how you know where you are in this conference room, how you know where you are within the context of this hotel, or even for me, Denver, since I'm from here and I have a good local map of the area. And we also try to understand how we use this sense of location or how to support memory processing. So decades of research have indicated that the same brain regions that are important for memory are also important for our sense of location or for navigation. And this is probably best exemplified by an experience that I'm sure we've all had of parking our car in a parking lot, going into work or a grocery store, and later not only having to remember where we parked our car, but having to accurately navigate back to that location. In order to successfully complete this task, what we really need is some sort of internal neural map of our external world. And we need this to not only know where we're currently located, but to know where we've been and to subsequently plan where we want to go. In order to do this, our brain has to integrate many different complex cues. For example, the brain needs to be able to recall and bring online a memory of where we parked our car, which is a multisensory representation. We also need to, as we navigate back towards our remembered car location, we need to be able to identify landmarks in the environment, the position of these landmarks relative to each other, as well as our position relative to these landmarks. We need to have information about the direction that we're traveling. Are we moving towards our car or away from our car? And we need to have some metric or understanding of the distance that we've actually traveled towards our goal location. So how does our brain accomplish this? Well, it turns out that our brain has neurons in regions called the medial entorhinal cortex, shown here in blue, and the hippocampus, shown here in orange, that contain all of the neural substrates that allow us to build an internal map of our external world. The first of these are called grid cells. They've been hypothesized to provide the brain's universal spatial metric system. This is an example of a grid cell shown here. What you're looking at is a bird's eye view of the activity pattern of an individual grid cell after a mouse has explored a 1 by 1 meter open arena for about 15 or 20 minutes, where blue colors indicate spatial positions where the cell was not active, and red and yellow colors indicate spatial positions where the cell fired at a high rate. Now, you can see even just by eye that in many ways this pattern is reminiscent of a longitude and latitude coordinate system, only instead of a square grid covering a map, the unit of the grid is an equilateral triangle. Now, there are two features of grid cells that allow them as a population to differ in important ways. The first is that different grid cells have different spatial phases. That means that they fire in different spatial locations in the environment. This is an example of that feature here, where you have two grid cells that have the same underlying geometric structure, but each one is slightly phase shifted from each other. They fire in slightly different spatial locations. And that means that no matter where you are in the environment, there's always some subpopulation of grid cells that are active. The second feature is that grid cells come in different spatial resolutions. So here's a sagittal slice of a mouse brain. And in dorsal entorhinal cortex in rodents, you'll find that the distance between these grid firing nodes is about 20 to 30 centimeters, while in ventral entorhinal cortex, you'll find that the distance between these firing nodes increases up to several meters. What this means is that you can take a relatively small number of grid cells, and by combining their information together, get a very accurate estimate of the animal's position in space. So grid cells coexist along several other cell types that are defined based on their firing patterns observed as animals navigate through the world. The first are border cells. These are neurons that fire only when the animal is near an environmental boundary. There are also head direction cells. which fire only when the animal faces one particular direction in the environment and are thought to provide a type of neural compass. And there are speed cells that increase or decrease their firing rate here on the y-axis relative to the animal's running speed here on the x-axis and are thought to provide a type of neural speedometer. There's one missing. There it is. And most recently, there's been object vector cells, which are cells that fire when the animals are a particular distance from an object in the environment. And together with border cells, these are thought to provide information about landmarks and boundaries in the environment. Together, then, as a population, these cells are thought to support behaviors like path integration-based spatial navigation, which is the ability of an animal to go from a starting point, take some sort of circuitous route out into the environment, and using only self-motion cues like optic flow or vestibular information, estimate their position in space, and use that estimate to calculate a vector back to their starting location. These cells are also thought to provide critical input to place cells in the hippocampus. These are neurons that fire only when the animal's in one particular place in the environment and are thought to provide the neural substrate for memory. Now, despite the really fascinating physiology of this system, one of the critical unanswered questions in the field has been, how does this neural system or these neural representations really support memory and navigation? And that's the question that I'm going to focus on today. So initial experiments proposed a dissociation or a possible dissociation between how representations in the entorhinal cortex versus the hippocampus might support memory and navigation. Entorhinal cortex was proposed to contain codes for spatial position, orientation, and running speed that were really independent of what we would call non-metric cues, cues like whether or not there's a reward in the environment or the behavioral state of the animal. On the other hand, place cells were thought to be sensitive to both metric cues, such as the geometry of the environment, as well as non-metric cues, such as the location of a remembered reward or goal position. This type of dissociation would allow you, for example, to encode, say, 4 different concerts that occurred in the same concert hall. So your hippocampal place cells would allow you to encode these as unique episodes or events, while your entorhinal cortex would allow you to have some kind of universal metric that would allow you to navigate across this single context. And more specifically, it was envisioned a little bit like this. This is just an illustration. Where, say, for example, each of these events, for each of these concert events, my place cells would remap in the sense that these place cell firing fields would change their firing rate. They would turn off, they would turn on, or they would move the location in which they're active. While my grid cells would remain relatively insensitive to these changes in what's actually going on in the environment and maintain this universal kind of metric that would allow me to navigate through space. It would only be if I attended a concert at a completely new concert hall that both my place cells and my grid cells would change and allow me to encode this as both a new episode and a new context. Place cells, again, would remap. They would change their firing rates or move positions, while grid cells would make changes like shifts in orientation, shown here, for example, or changes in the distance between these firing nodes. However, subsequent work started to reveal that entorhinal codes are actually sensitive to a range of different cues in the environment. And the first sort of set of work that really looked at this came from studies showing that entorhinal codes are sensitive to changes in environmental geometry. And this was first illustrated in some earlier papers here, and most recently we replicated this in mice. And the basic paradigm demonstrating this first allows an animal to explore a 1 by 1 meter open arena, such as a square, for example, until they become very familiar with the environment. You then introduce, for example, a wall that makes this environment no longer a square, but instead an asymmetric rectangle, and then look at what happens to grid cell firing fields. And in this case, what you find is that grid cells will actually rescale, so the distance between their firing nodes will actually get smaller only along the axis that was modified. So in this case, this axis here, whereas on the axis that's not modified, they'll remain relatively stable. It's not just grid cells that are sensitive to the geometry of the environment. Speed cells are also sensitive to these changes. This is an example of that phenomenon here, where speed cells will change their firing rate by running speed relationship when you change the geometry of the environment. Here you can see the firing rate of this cell on the y-axis and the running speed of the animal on the x-axis. And in the modified environment, you can see that, the slope of that relationship has increased. And head direction cells will also shift their orientation under these conditions to align with the modified axis, or at least a subset of head direction cells will. So here I'm showing you 2 head direction cells and their tuning curves in the baseline condition in purple and in the modified condition in light blue. And you can see that between these 2 environmental geometries, there's actually been a shift in where the cell prefers to fire. However, it's not just environmental geometry. It turns out that entorhinal representations are also very sensitive to changes in the animal's behavior. And we recently showed this by looking at a very simple change in the animal's behavior, their running speed. So we looked at what entorhinal representations were when animals were running fast, say over 10 centimeters per second, versus when they were running slow, not stationary, but running slow. And what we found was that first, the number of neurons in entorhinal cortex that actually encode information about the animal's position, running speed, or orientation actually increase when the animal's running fast. So for example, when the animal's running slow, there are fewer cells that actually encode information about the animal's position compared to when the animal's running fast. And in addition, we see more what's called mixed selectivity when the animal's running fast, meaning that neurons encode for more than one variable, one navigational variable when the animal's running fast. So for example, when the animal's running slow, you might have a neuron that's, say, only tuned or encoding the position of the animal, but when the animal's running fast, this neuron might encode both the position and the head direction of the animal. And this actually has some important implications for how these codes can be decoded by downstream regions. It turns out that much of this improvement in coding is due to a sort of amplification or increase in the signal-to-noise ratio of these cells. So here, for example, is a cell that when the animal's running slow, just very weakly encodes the position of the animal. And when the animal's running fast, you can see that a more coherent and clear representation for the animal's position emerges. And so this indicated to us that entorhinal neurons can actually vary their tuning properties based on an animal's behavior. And this finding then really led us to consider, do entorhinal neurons or do entorhinal codes potentially adapt under other types of task conditions? And our motivation was that many ethologically relevant navigational strategies often employ a variety of different types of navigational strategies. For example, goal-directed navigation to remembered reward locations. And up until this point, much of the work looking at entorhinal neural representations during navigation, not all, but a vast majority of it, had used just a single behavioral paradigm. And that behavioral paradigm is to allow a rodent to randomly explore for randomly scattered food rewards in a large open arena. It's sometimes called the Hoover task because rats kind of just lick up the Cheerio dust as they move around this environment. But we know that many animals do not just randomly forage for freely available food sources. They often navigate in a manner in which they are trying to go back to a remembered goal location, such as a food location or a nest location. Indeed, this is often how I navigate in order to reach my, I don't just randomly navigate, I in fact navigate with a goal in mind. And so we wanted to ask, are neural codes and entorhinal cortex actually influenced by tasks that might integrate a remembered reward location? And to look at this, we designed the following task. So we had 2 boxes, this black box here, and then a second box, which was white. And these 2 boxes are in the same spatial position, so we would literally just lift the black box out and place the white box down. When the animals are in the black box, they're performing this oft-used random foraging task, so they're just foraging for crushed-up Cheerios that are scattered in the environment. And when the animals are in this white box, they actually switch back and forth, so they randomly forage, but when they hear an auditory tone come on, they have to go back to an unmarked remembered reward location, which I've indicated here in red, in order to receive a large food reward, an entire Cheerio. And animals actually learn this task very quickly. These are rats. So here's an example of an animal's behavior in the free foraging task. The trajectory of the animal is shown in gray, and this is where the remembered reward location will be in environment 2. And you can see the animal covers the space quite well. And then here in environment 2, I'm showing the random foraging trajectories in gray here, And then individual example trajectories from when the tone turned on to when the animal entered the reward zone are shown in different colors. And you can see that animals take relatively direct paths back to that remembered reward location. We're then going to look at grid cells, border cells, and head direction cells, as well as what I'll call non-grid spatial cells. These are neurons that encode the position of the animal, but they don't do so in a geometric way. They don't have a grid cell firing pattern or a border cell firing pattern. And we're going to look at these firing patterns not just in the animals that are performing the task in environment 2, but critically we're going to compare all of our data to a control set of animals that experience environment 1 and environment 2, as well as the auditory tone, but they don't have to perform any task in environment 2. They're just going to be randomly foraging. Okay, so we first considered grid cells. This is an example of 3 different grid cells, each recorded in environment 1, where the animal's randomly foraging, and then recorded in environment 2, where the animal's performing the task. And the first feature of the grid cells that we looked at was the spacing, so just the distance between these grid firing nodes. And here what I'm showing you on the top plot is the grid spacing for experimental animals in environment 1 on the x-axis, and then in environment 2 on the y-axis. And what you can see is that there's been this small but significant shift in grid spacing, such that the spacing is smaller in environment 2 compared to environment 1. This is shown here again compared to what we see in the control animals. So on the x-axis is the change in grid spacing between the 2 environments, and on the y-axis is the number of cells that we observed to show each measurement. And you can see that in the experimental animals here in red, there's been a small but significant decrease in grid spacing, whereas in the control animals, we didn't actually see any significant change in grid spacing between the 2 environments. We next looked at orientation. So this is just the tilt of the grid pattern. I've highlighted that here with these white lines. And here what we saw was even more striking. So here on the x-axis is the change in grid orientation between environment 1 and environment 2. And you can see that in red, most of our experimental animals showed a dramatic shift in grid orientation between the 2 environments. Whereas in the control animals here in gray, we didn't really see any significant changes in grid orientation between the 2 environments. Because the grid cell shows 60-degree symmetry, this bar plot here at 60 is in essence 0. So much of the grid cells in the control animals maintained their representation across the 2 environments in terms of orientation. So what does this mean? This means that despite the fact that these 2 environments share the same underlying geometric structure, we see signatures of contextual remapping in the presence of a task. It's not just grid cells that were sensitive to the presence of a task. We also saw remapping or changes in the head direction cell system. So here what I'm showing you are 4 head direction cells, and their tuning curve in environment 1 in blue, and their tuning curve in environment 2 in red. And you can see from these latter 3 examples that in environment 2, the orientation or preferred location in which the head direction cell is firing actually shifts between these 2 environments. And interestingly, if we look at the change in orientation in our head direction cells here on the y-axis versus the change in orientation in our grid cells here on the x-axis, you can see that there's actually a tight correlation. So the amount of rotation that we're observing in the grid cells within sessions or within animals is actually highly correlated to the amount of rotation that we see in the head direction cell system. We also see changes in border cells. I think this middle example here is probably one of the most striking examples that we observed, where you have a border cell in the environment 1 that fires very strongly on the east wall, and in environment 2, it fires very strongly on the north wall. If we look at all of the border cells, we find that there is a population that remains stable between the 2 environments, but there's also a very substantial population that shifts by 90 degrees and even a few border cells that shifted by 180 degrees. And I should note that we didn't see any of these changes in border or head direction cells, broadly speaking, in our control animals that experienced both environments but no change in task demand. We also saw changes in these non-grid spatial cells. Non-grid spatial cells are known to be sensitive to changes in things like the color of the box, but we saw significantly more remapping in our non-grid spatial cells in our experimental animals compared to our control animals. Okay, so we next wanted to ask, do these entorhinal maps restructure in a manner that potentially incorporates information about the remembered reward location? So we first looked at grid cells, and what I'm showing you here is the firing rate of grid cells on the y-axis relative to the position of the animal, or how close the animal is to the reward location on the x-axis, where 0 indicates that the animal's at the reward location, and 100 indicates that the animal's as far from the reward location as it could be. And then I'm showing you the firing rate of grid cells in environment 2 in red and in environment 1 in blue. And this gray curve is just the difference between those two lines. And I think what you'll see is that when the animal's near the reward zone in environment 2, there's actually a significant increase in the firing rate of grid cells. We did a variety of analyses to look at why this was happening, and I'm just going to summarize them here with this illustration. It turns out that what's going on is that when the grid cells are in, or when the animal's in environment 2, whatever grid nodes are closest to the reward zone increase their firing rate, whereas the nodes that are further from the reward zone either stay the same in terms of firing rate or decrease their firing rate. And so this indicates to us that, generally speaking, the relative activity of grid cells is increasing near the reward zone only in environment 2. We saw the same thing in these non-grid spatial cells, so I'm just showing you that here. It's the same plot that I showed above for grid cells. This effect in the non-grid spatial cells was due to a variety of changes in their firing structure. I'm just going to highlight 2 of those changes that I think are particularly interesting. In the first, we saw some cells that would actually shift their firing field to align with the reward location. I'm showing that 2 examples of that here on the top row. You can see a cell, for example, that has a coherent, nice, spatially tuned firing field in environment 1, and this firing field actually gets much closer to the reward zone than expected by chance in environment 2, and the same for this cell here. We also saw another subset of cells that I thought was very interesting, in which they had a sort of less coherent spatial firing pattern in environment 1, but in environment 2, they would have this very tightly tuned, spatially coherent field that emerged over the reward zone. And so while non-grid spatial cells remapped in a really heterogeneous manner together as a population, what this resulted in an increase in activity near the reward zone in environment 2. Okay, finally we asked, how might task-associated changes in entorhinal cortex impact navigation? And to look at this question, we estimated the animal's position from simultaneously recorded cells. We had a subset of sessions in which we recorded many tens of cells, which allowed us to perform this analysis. And we trained a Bayesian decoder on position and speed matched data from environment 1 and environment 2. And what we found was, this is for an individual session, so this is just an example session. What we found was that we could decode the position of the animal to a much better degree, shown here as the amount of error on the y-axis, when the animal was near the reward zone in environment 2 compared to environment 1. And this is for all of the sessions combined, so you can see it quite clearly here, where when the animal's in environment 2, we actually can decode the position of the animal better near the reward zone compared to environment 1. And here, this is just the difference between the 2 curves. So what this tells us is that these localized changes in firing rate that I described to you on the previous slide result in improved spatial decoding near the remembered reward location. So let me just then summarize this first part of my talk. I showed you that entorhinal maps are sensitive to environmental geometry as well as running speed and potentially the behavioral state of the animal. And I also showed you that remembered reward locations can serve as a contextual cue capable of evoking different entorhinal maps. And there was also a very nice second paper that was published alongside of ours that I'd encourage you to take a look at, that looked at how entorhinal maps restructure in the presence of a remembered, or in the presence of a goal-directed navigational task. And it's just kind of a take-home message. The way we sort of envision this is that you could have a mouse that, say, navigates through this local environment. It's very familiar with the environment. This is the city zoo in New York City. And it randomly finds food on occasion. But one day there's a really awesome pizza party at the penguin display. And it might actually benefit this animal to create a new map of the same spatial context that perhaps incorporates or emphasizes information about that particular reward location. And more broadly speaking, these entorhinal representations that I've described to you have now been found in a wide range of species, from the goldfish and fruit fly to bats, rodents, non-human primates, and primates. And so we think they really represent an evolutionarily conserved coding scheme for navigation. And the fact that entorhinal cortex can dynamically alter its coding features to integrate contextual features, we think would allow it then to support a really wide range of navigational strategies. Okay, so in the last part of my talk, I just want to briefly tell you a little bit more about how we're thinking about remapping. I just told you that we found unexpected features of an animal's environment, behavior, and experience that can drive remapping in entorhinal maps of space. But what are the principles that really govern the formation of new maps of space, and how might we go about identifying these principles? Well, to consider this question, we turn to place cells in the hippocampus. So why the hippocampus? There have been decades of really significant and beautiful work that have looked at how hippocampal place cells can robustly remap. And by remap, I mean that place cell firing fields between environmental contexts will turn on, turn off, change their firing rate, or move the spatial position in which they're active. We know, for example, that place cell remapping is highly sensitive to many features, such as different rooms. So for example, if you have the same black box in two different rooms, you see a lot of place cell remapping. We also know that place cell remapping is sensitive to environmental geometry. So between, for example, a square and a circular box, you'll see a lot of remapping in the place code. We also know that experience plays a role, that remapping patterns can change as animals learn about the nature of different contexts. And in particular, experience seems to be critically important for the remapping patterns and dynamics that are observed when animals experience environments with many overlapping sensory features. For example, you could imagine a rodent becomes very familiar with these two different backyards. And later, when it encounters some sort of hybrid backyard that incorporates overlapping features of both of these yards, it has to decide whether or not to encode this as a new context or identify it as perhaps one of these contexts here. And this could have critical implications for survival if, for example, this first backyard is associated with freely available food and the second backyard is associated with a predator like a cat. So the remapping dynamics that are observed in the hippocampus under such conditions have probably been best studied in a task in which they used a morphing experiment. So in this task, these are freely moving animals. They experienced a square box and a circle box, as well as a variety of squircle boxes. So these kind of hybrids between square and circle boxes. When you look at this type of morphing task, various experimental protocols can actually result in very different findings. So for example, in one paper, for these squircle boxes, they found that, excuse me, that hippocampal neurons will either vote that the animal's in the square box or the circle box. It won't really create representations of these sort of in-between environments. Where in other cases, you can have this kind of gradual shift in the representation in the hippocampus between the square and the circle. And there was some really recent nice theoretical work that was posted on bioRxiv that proposed that the effects that these effects can empirically reflect the experience of the animal and be observed in any animal. And it's really a reflection of what the animal has subjectively learned about the likelihood of experiencing particular contexts. And so we wanted to look at this question directly and ask, does an animal's prior beliefs about the frequency with which it encounters stimuli determine the remapping patterns that we would observe in the CA1 region of the hippocampus? And so to do this, we performed 2-photon imaging, which allowed us to look at between 200 and 2,000 CA1 cells simultaneously. And we designed a virtual reality version of the morph task, so I'm showing you that here. We changed 3 features of the environment: the orientation and spacing of these bars on the walls, the color of the towers here at the start of the track, and then the contrast of this of the sky that the animal can view. And this morph task went from 100, so this is one context here, to 0, which is a second context here. And then we could create continuously representations between these two extremes. We then had 2 training protocols. So we had a rare morph condition in which the animal primarily only experienced morph 0 or morph 100 for the course of 7 days. And then on day 8 and beyond, it would experience all of these environments. Or we had a frequent morph condition in which the animal experienced all of these environments every single day. And the data that I'm going to show you today comes from these imaging sessions from day 8 and beyond. Okay, so can we quantify what we might expect to see if remapping is driven by prior beliefs? So in this case, again, I'm just going to remind you we have these two training conditions, the rare morph condition and the frequent morph condition. For the rare morph condition, if it is the case that remapping is driven by prior beliefs, we would predict that the animal would have some sort of bimodal prior over morph values, which I'm illustrating here, where P of S is the prior distribution and describes the frequency of previously observed stimuli, and morph value here is on the x-axis. Whereas for the frequent morph, the animal would have a relatively flat prior across the different morph values. What this means is that an observer that's performing probabilistic inference would make different predictions about the identity of these intermediate stimuli. And we can quantify this using map inference, which is just the process of taking the maximum value from the posterior distribution using Bayes' formula. So in the case of the rare morph condition, what we would predict is a discontinuous estimate. So that means that CA1 would essentially vote that the animal's in context 1, or morph 0, or context 2, or morph 100%, when the animal encounters these intermediate environments. Whereas for the frequent morph condition, we would predict that we would see some sort of gradual or continuous shift in the representation for these in-between environments. So what do we actually see in the data? Well, first, this is just an example imaging session from a rare morph mouse and a frequent morph mouse. And what I'm showing you here is the activity of these individual place cells that have been sorted based on the location on the track in which they're most active. So here, the position of the animal in this case is on the x-axis, and then these cells here are stacked on the y-axis. And so first what we found was that there was a large population of cells that showed place coding in both the rare morph and the frequent morph condition. So I'm showing you that here. This is the 0 morph condition. And I'm now going to keep the sorting that we've performed for this 0% morph condition across the other morph conditions. And I've just binned them here into these 25% values. And what you can see is that if we move to this extreme example of 100% morph, there's been a complete remapping in both the rare morph and the frequent morph. But there are still place codes in this 100% morph context. So if we again sort the activity of cells in the 100% morph, you can see that there's this nice representation of the entire track in both the frequent and the rare morph conditions. But if we carry that sorting back to the 0% condition, you can see that that gradually fades out. But what I hope you'll appreciate, even just from seeing this single session, is that there's a very different effect for these in-between contexts or environments. So here in the rare morph, there's a really clear and discrete shift around 50% to a new representation, whereas in this frequent morph, there's kind of a fade out and a fade in of the representation. And this is even clearer if we look at all of our sessions combined. So this is all of our cells imaged together. And again, you can see this really discrete transition in the rare morph condition and a more gradual fade out and fade in for the frequent morph condition. We can look at this another way or quantify it further by constructing trial-by-trial population similarity matrices. So in this case, what we're taking the activity of the entire vector of CA1 cells that we're recording from. I'm showing you an example of 3 CA1 cells imaged here. You can see the morph value on the y-axis, so this is sorted by which environment the animal is experiencing, and on the x-axis is the position of the animal along the VR track. And what we can then look at is what is the correlation of the population of activity for, say, 0% morph versus another morph percent, say 10 or 15%, and that correlation in the rare morph condition is very high, as indicated by this black color. But if we look at another set of morph values, so say we look at 5% morph condition versus a much later morph condition, say 75%, you can see the correlation is very low, as indicated by these white values, or white color. And this blocky structure in the rare morph condition just again quantifies what I showed you on the previous slide, that around this 50%, 40% mark, you see this discrete transition to a new representation. If we look at the frequent morph condition, you can again see that there's this kind of fade in and fade out of the representation that's relatively continuous across the morph values. Here, this is for an individual session from a rare morph, an individual mouse from a rare morph condition and a frequent morph condition. This is all of the mice combined. We've binned these trial-by-trial similarity matrices, and again, you can see the same effect that I've described on the previous slides. Okay, and then finally, I just want to compare what we actually observed experimentally to what we sort of predicted would happen. So here again, this is kind of our analytical or computational predictions for what we might see in the rare morph versus the frequent morph. And we can actually compare these predictions to the experimental data by just calculating the similarity fraction on the population vector of hippocampal activity on a single trial and comparing it to the activity of the hippocampal population on morph condition 0, which is 0%, or 1, which is 100%. And just to kind of give you a quick layout of what these graphs actually mean, these higher values are going to mean that the CA1 population shows a representation that's more similar to what we see at the 0% morph condition, and these lower numbers indicate that it's a representation that's more similar to what we see in the morph 100% or 1 condition. And here, an individual are shown in different colors. All of the individual animals in different trials are individual dots. And then I've overlaid these predictions here, and you can see that there's actually a really nice match between kind of what we would predict theoretically and what we see experimentally. Okay, so just to summarize, I showed you that you observe this voting behavior in the rare morph condition, and you see more gradual changes in CA1 representations in the familiar morph conditions. And that CA1 representations really do appear to reflect the statistics of the animal's experience. And I think it's important to remember that natural stimuli and our experience of context as we walk through the world is really continuous in nature. So this is me walking between 2 buildings in Stanford. And what the hippocampus really has to do is be able to take these continuous representations and decide when to chunk them into individual episodes or different contexts. And we think that looking at CA1 remapping dynamics using the kinds of tasks that I've just described to you can really tell us something about how the hippocampus is able to take continuous events and chunk them into discrete memory episodes. And finally, I just want to acknowledge and thank the amazing people that did all of the work that I presented today. So this is the lab as well as previous lab members and many of our collaborators. Rob Munn is a really fantastic, he was a postdoc in the lab, he's now a PI at the University of Ireland in Galway. He performed the work looking at how environmental geometry influences entorhinal representations. Kia Hardcastle and then our collaborators Surya and Niru looked at how behavior, the animal's running speed can influence entorhinal representations. Will Butler and Kia were also the lead authors looking at how remembered reward locations can influence entorhinal representations. And then finally, Mark Plitt did all of the imaging work that I showed you in the latter part of the talk. And I'd like to thank my funding agencies as well as all of you for your attention. Thank you.
Okay, we have time for a few questions.
Good morning.
Hi, Dory.
I'm sort of wondering, so when you have the reward inside the environment, and as a result, the grid field moves towards the reward or moves a bit in the environment, how does that work with the idea that grid cells are used for navigation and you have this nice grid organization? I think these things are not working together very well.
Yeah, so I would just clarify that we didn't actually observe the grid field moving. The Baccarat paper from the Churchvari lab did observe something more like that. It was a slightly different task. The animal had to learn different reward locations each day. What we saw was just an increase in the firing rate. So in that sense, that is a little bit more consistent with kind of the viewpoint of grid cells serving as this sort of universal metric. But I think that grid cells clearly can shift their firing fields depending on the demands of the task. How that actually fits in with this idea that they provide like a universal metric of space, I think is something that we're still trying to get our hands around, yeah.
Thanks.
Great question, yeah.
Hi.
Hi.
Thanks for the Thanks for a very nice talk. Just for your experiment before frequent morph condition, I think like brain can represent the categorical signals if they are required to. So I wonder like if you train the rats to like make it sort of binary decisions to act this way or that way, like based on the, like what the morph is, then you might start to see more like binary representation compared to gradual, even if it's in a frequent condition.
Yeah, that's a great question. I didn't present it, but we did actually try that. So we had a task in which the animal had to lick at tower 1, let's say, if they were in context 0% through 49.9%, and they had to lick at tower 2 if they were above that. And that didn't seem to drive discretization in the representation or categorization. That being said, it could be that, if you had a different type of task, you could potentially see something different. But we found that these remapping dynamics are very robust to task demands. So those were mice that were kind of trained in the familiar morph condition and then were performing this task. But it's a really good question, yeah.
Have you done this experiment where you morph back from 1 to 0 and see if the place field will recover to the original field for gradual versus the jump?
Yeah, so all of the morphs are randomly interleaved. So they're not like 0 to, maybe the color bar kind of indicated that, but they're actually randomly interleaved. So the animal on any given trial can experience any morph percent value when we're actually doing all of the different environments.
Okay.
Yeah. Thank you.
Hi. I wonder what was the dynamics of entorhinal changes during the morph, and then what would be your predictions about the relationship of that with the place field changes?
Yeah, that's a great question. We have not looked at entorhinal cortex during that task. I think that would be a really interesting thing to do. I would suspect that it might show something somewhat similar, just given that it seems to be strongly driven and provide a lot of input into the hippocampus, but I don't really know. I think that would be, that's something we're interested in looking at in the future. Thank you.
So I have a question. In the first part of the talk, in the Butler et al. Work, do you have any idea whether disruption of activity in the entorhinal cortex would affect behavior and the performance?
You mean
in the reward? We haven't done that experiment, but I think it'd be really interesting to look at. My guess would be that they might still be able to perform the task, but I would expect to maybe see some kind of deficit. Maybe they would take longer paths to the remembered reward location. We have shown that if you perturb the spatial frequency of the grid cells, so if you make the grid cells have larger distances between their firing nodes, animals can't remember multiple reward locations within a single spatial context. So it could be maybe they could perform the task I described, but they might have more difficulty doing the task in the other paper, the Baccarat paper, where you have to remember new goal locations each day. Yeah. Thank you. Okay. Let's thank Lisa again. Okay. The next talk is by Marcella Noorman from Janelia Research Campus.
Thank you very much for this opportunity. So Lisa just gave a beautiful talk on the challenges of navigation, which is a function that many animals, from rodents to insects, have to perform. Fruit flies also navigate and can do so even in the absence of visual or olfactory cues. So for example, in this infrared imaging video, you can see a hungry fly, which is in complete darkness, that has just encountered a spot of odorless food and is able to repeatedly navigate back to this same spot without any guiding cues. Now, in the fly, the ability to flexibly navigate is thought to rely on this central brain region called the central complex. Within the central complex, this donut-shaped structure, called the ellipsoid body, contains neurons that are tuned to the fly's heading. As you can see from this EM-based reconstruction, the neural processes of these neurons tile this entire donut. Now, if we monitor these neurons while the fly is walking in darkness, you can see that they maintain a localized bump of activity that tracks the fly's heading. Now, this requires that this bump both persists when the fly is not turning, as it is here, and also that this bump updates accurately when the fly does turn, as it does here. So for over 2 decades, theorists have used ring attractor networks to model similar systems in mammalian brains, such as the head direction system. So let's arrange our neurons in a ring according to their tuning to different headings. So theory suggests that to maintain a stationary but persistent bump of activity, neurons with similar tuning should locally excite each other, while neurons with different tuning should be silenced through broad inhibition. Now, to move this bump of activity when the animal turns, theorists have suggested the addition of two side rings that inherit the bump of activity from the center ring and then have shifted connections back to the center ring. So, if the animal turns clockwise, one side ring becomes more activated than the other and shifts the center bump counterclockwise. The other side ring operates in a mirror symmetric way when the fly turns in the opposite direction. Now, the fly brain has a structure that behaves much like these side rings. So either side of this handlebar structure, called the protocerebral bridge, gets either clockwise or counterclockwise angular velocity information and uses this information to shift the bump of activity when the fly turns. So you can think of the sides of the bridge as being the side rings. However, there is one key difference between the fly circuit and classic ring attractors. Classic models assume you have sufficiently many neurons to approximate the system as continuous, which is done mathematically by taking the number of neurons to infinity. Here, however, we do not have enough neurons to make this assumption. So as you can see from this side view of the bridge, there are actually only 8 discrete computational units. So how then would a discrete version of this model with only a handful of neurons perform? Well, it turns out that this discrete model suffers from failures that the fly circuit does not. So now we're going to walk through those failures, and we'll compare them to data from a single fly walking on a ball in darkness. To start, let's see how the model would respond to a constant input velocity. Now, if we had infinitely many neurons, the system would perfectly integrate this constant velocity, and so the bump position would change linearly over time. When we only have a handful of neurons, and here I'm showing you 6, we see this is not the case, and rather the bump position traces out this wavy trajectory. Now, unfortunately, we can't just ask a fly to turn at a constant angular velocity for 10 seconds to compare to this aspect of the model. But what we can do is measure the average angular velocity of the bump over a small time window. And if we do this when the fly is turning at many different angular velocities, we see a pretty tight match. Our model, on the other hand, performs much worse. And in particular, you can see that for these smaller angular velocities, the model bump gets stuck where the fruit fly appears to do just fine. Okay, so now what would happen if the fly were to stop moving? Well, if we track how much the bump drifts after the fly stops, we end up with this distribution that is peaked at 0. So this indicates that the bump does not tend to drift much. For our model, however, this distribution of drift is more broad and uniform, indicating that the bump does tend to continue moving after we've shut off the input velocity. And indeed, if we go back to our simulation, we can see this. So if we turn off the input velocity, we'll see the bump drift before stopping. Okay, so clearly this model suffers from problems that the fly circuit does not. Now, to understand why it performs so badly, we're going to dissect it analytically. So when we do this, we're going to characterize this bump of population activity by its position, or where it sits around the ring, its width, and its amplitude. All of these quantities are coupled and will change in time. These quantities are also shaped by the maximal strength of the local excitation in the system, JE, and the strength of the broad inhibition, JI. So to start with, let's see what happens in the case when we have, in the absence of input velocity, when, as you'll recall, the model bump continues to move. So we can actually show analytically that in the absence of input velocity, the bump's velocity actually depends on both the width of the bump and where it's located around the ring. Now, I'll visualize this function for you in a moment, but I want you to keep in mind that when this function is 0, which I'll indicate by white, the bump can be stationary. When this function is positive, indicated by red, the bump will be pushed clockwise, and when this function is negative, or blue, the bump will be driven counterclockwise. Now, if we had infinitely many neurons, this function actually becomes 0 everywhere, this means that the bump can stop anywhere without drift. When we only have a few neurons, however, this is not the case. So I'll unveil these hatched regions for you in a moment, but first I just want you to notice that for most of this space, there are only a finite number of places where the bump can sit, and only half of these places are actually stable. So as you might have guessed, since I'm hiding these regions from you, there are places where things are better. So for example, if your bump width is pi, then it turns out the bump can sit anywhere around the ring. But this requires that your bump width stop changing as well. And in order for this to occur, we can again show analytically that there is another relationship between the bump width and position that must be satisfied. And this relationship is actually determined by that excitation parameter, JE. So different values of this parameter determine different curves in this space. And if we, for example, select this value for our parameter, that means the system must be on this contour in order for the bump width to stop changing. Okay, so it turns out there are two constraints that must be satisfied in order for the bump to be stationary. First, you must be on a zero or white part of this function, and note this does not depend at all upon your parameters. And second, you must be on the appropriate contour that corresponds to your excitation. So for example, this one. Now, let's imagine that the system were at this point when the input velocity was turned off. As you can see, even though we are on a zero of this function on the left, we are not on the correct contour over here on the right. And so because of this, the bump width must change. But when the bump width changes, we have now moved off of the zero for the function on the left, and so the bump position must change. And this will continue until the bump settles at a point where the zeros of this function line up with the appropriate contour. Okay, so now let's see what happens if we consider many different contours and we overlay them with this function. Well, as you can see, it turns out that some of these contours actually line up perfectly with the zeros of this function. So this means that if you select your excitation carefully so that you end up on one of these three special contours shown here in black, then the bump can actually be stationary at any point along the ring. Note that this means that the width of the stationary bump will depend on where you are on the ring, and you can think of these contours as basically trading the oscillation in the bump velocity for an oscillation in the width. Okay. So now let's think about what would happen if we drive the system with some non-zero input velocity. Well, ideally, you would stay near these special contours. If you move off of these contours, you will get an oscillating velocity contribution from the alternating red and blue regions. Now, while you can't always perfectly stay on these contours, it turns out that if you start out on one, you will tend to stay close to it. So to see this, we can simulate the system while driving it with a constant input velocity. So this plot on the top will show you the actual bump of activity. The vertical dotted lines indicate the tuning or the preferred headings of the individual neurons in the network, and the y-axis will show their activity. This green circle represents the center of the population activity, so this is our bump position. Now, if the system were to integrate this constant velocity perfectly, it would perfectly track this white dot here. You can also see this in the bottom right corner, where we'll trace out the bump position over time, and this is identical to the plot you saw earlier. So to start with, I want you to pay attention to how this bump moves. As you can see, it has this kind of chugging movement, which is caused by the bump slowing down and speeding up. And now, if we turn off our input velocity, you can see that the bump actually moves backwards. So in order to understand why this occurs, I want you to pay attention to where we are in this function. So as you can see, we're not starting on one of those special contours. And as the bump moves, the blue regions, which are pushing the bump counterclockwise, are causing it to slow down, while the red regions are causing it to speed up. And now, when we turn off our input velocity, the bump is in a blue region, so rather than stopping, it's pushed backwards. Okay, so to compare, let's see what would happen if we select our excitation parameter so that we do start on one of those special contours. So here, the blue and the purple bumps should do much better. And indeed, as you can see, they do move much more smoothly, and this is because they're staying near the white parts of this function. And now, when they're supposed to stop, they actually do. Okay, so in summary, I've shown you that a small circuit can integrate angular velocity just as well as a large circuit can, though you have to select your local excitation more carefully. Moving forward, we're using the recently released EM connectome of the fly brain to identify neural circuit mechanisms that might be able to properly tune the local excitation in the system. And with genetic access to the to the neurons in this circuit, we can test these mechanisms experimentally to uncover whether or not the fly's heading system is tuned in the way this theory suggests, or if it's using an entirely different solution to overcome the limitation of being a small circuit. Together, this provides exciting new directions for understanding how brains, even those of small animals, generate accurate internal representations of where they are in the world. And with that, I would like to thank a number of people, but in particular, my PI, Anne Hermundstad, and Sandro Romani, and Vivek Jayaraman, and all of you for listening. Thank you.
So the beautiful analysis that you showed was in terms of the drift velocity as a function of position and the width. Mm-hmm. I was wondering whether it's possible to analyze, or whether you try to analyze a system in terms of an energy function, a Lyapunov function that would need to be smooth, flat, in order to avoid the drift.
Yes, that is exactly kind of what is occurring there. Is you move from these situations where you kind of have these valleys in your energy function where there's these discrete attractors, and this basically smooths out and you get a flat actual ring attractor back, even though it's a discrete system.
Very nice. I'm wondering, is there any input of the absolute visual position in order to, on occasion, register the attractor to the right direction from outside?
Not in the model I showed. But the fly circuit does get visual information that we think helps kind of error correct and also help stabilize the bump.
Hi. In order to test the theory, how do you think you can do that by experimentally, like changing the synaptic strength? Or?
So what we're kind of looking at right now, and in this Turner-Evans paper on bioRxiv, they found in the EM, and this paper is using a partial connectome. They found in the connectome many more recurrent local and hyperlocal recurrent loops than originally thought to be there. So the first thing we kind of want to do is now that we have this full connectome, kind of verify those recurrent loops, see if we can determine kind of what we expect the effective excitation kernel to be. And then the next steps would be experimentally kind of perturbing these things, but we're not quite sure how we'll do that yet. Thank you.
Any other questions? Okay, so let's thank Marcella. And the next speaker is McKenzie from NYU.
All right, thank you very much. It's my first time at Cosyne. It's been a fantastic meeting. Today I'm going to talk about how is it that pre-existing structure and dynamics in hippocampal circuits constrains the kind of plasticity that one can induce experimentally. So if we consider this classical theory of the hippocampal contribution to episodic memory, the index theory, states that when we have a new experience, there's a constellation of different sensory stimuli that define that experience and activates different patterns of activity in the cortex, which in turn will make some neurons in the hippocampus be active and some silent. And so when this pattern in the hippocampus is learned, it's said to be a so-called engram, and sets an index for the pattern of activity in the cortex that was active at the time of learning. Now, when that pattern of hippocampal activity is reinstated at a subsequent time, the idea is that that can reinstate the pattern of activity in these cortical modules which were active at the time of learning, and therefore we have the subjective experience of recollection of that episodic memory. And so if you take this kind of theory seriously, it becomes very important, how is it that different patterns of the hippocampus get bound to the afferent inputs that define a particular episode? How is it that the engram is imprinted, so to speak? And so a beautiful set of experiments got at this kind of question. And here what was done was the animal is head fixed, running on a track, and current is injected intracellularly inside of a single neuron. And here this example cell is shown here. And what's shown on the x-axis is the position of the animal on the track. What's shown on the y-axis is trials. And you can see that this neuron is silent before the current is injected. Current is injected, inducing these plateau potentials. Then the current is not injected anymore, and you can see that now this neuron continues to fire in the same position, as though now this neuron is bound to the afferent inputs that define this position on the track. And what's quite striking about this is even though the current was only injected for a limited amount of time and space, the neuron continues to fire over a rather broad region of the track, suggesting that the depolarization of the postsynaptic cell was able to bind afferents that occurred over order of seconds before and after the cell was depolarized. So this work was done by Jeff McGee at Genelia. There's also nice work with juxtacellular stimulation showing similar kinds of plasticity for hippocampal place cells. And so one of the conclusions that one could draw from this kind of work is that there's an omnipotentiality in the hippocampus where sufficient depolarization could now bind any neuron to any pattern of afferent inputs. But I believe, probably like most of the people here, that these hippocampal indices are not single neurons, but groups of neurons firing in particular patterns. And so now the technology exists where we can actually drive small groups of neurons to see how they get bound to the sensory inputs that define particular regions in space or particular episodes. So what we did is we turned to the technology of the silicon probes with LEDs microfabricated amongst the recording sites. So the LEDs are tiny, and so we can stimulate very small groups of neurons at fixed positions in space as animals are freely behaving. So this is data from CA1. We can change the intensity of the LED to parametrically change the average firing rate of the neurons recorded on that shank. And the neurons that are recorded only 250 microns away don't show any changes in firing rate. So we can get quite focal optogenetic stimulation here in a time-resolved manner. So we can reproduce the kind of experiment that's been done in a head-fixed situation, where you can see if I'm stimulating, in this case, shank 3. I can depolarize small groups of neurons as the animal crosses a fixed region in physical space as it's running back and forth on a linear track for water reward, and not modulate the firing rate of neighboring neurons simultaneously recorded. So what happens in these experiments? So this is data from a single CA1 neuron. On the x-axis is position, on the y-axis is trials. And the way that I set up my task is I run a baseline set of trials to see where the place fields are. I stimulate, I turn off my stimulation to see how the receptive fields are stable. I put the animal back into its home cage, and then I bring the animal back to the linear track in the afternoon to see how stable those receptive field changes were. And so in this case, when I depolarize a neuron and I turn off my light, you see that there's one place field that's stable throughout, and there's another place field which is induced. And so this is kind of like the finger of God experiment that Jeff McGee was doing, where he can depolarize a neuron anywhere, and the neuron will continue to fire in that place, suggesting an omnipotentiality. But in my hands, this was the exception. I saw other neurons that look like this, where they'll have a place field, I'll depolarize, and then I see remapping. And this remapping can actually be anywhere on the track. It doesn't seem to be tied to the location in which I depolarized. And then I saw other very funny things. So in this case, this neuron is not actually being depolarized by the light. This is, I'm stimulating a neighboring shank. It has a stable place field throughout, but I also see reorganization. And so what is going on here? All of this data is taken from area CA1, where you don't see very strong excitatory recurrence between these pyramidal cells, suggesting that perhaps any kind of change that you see in these non-stimulated cells here is likely due to a circuit effect. So we just want to quantify this ray mapping, and so what we did is we built a template for what the place cell activity looked like prior to the stimulation, and then we correlated that template with the activity trial by trial, obviously leaving out that particular trial and defining the template. And for those cells that remap, you would expect to see that the correlation should be high in the beginning, and then when the cell remaps, the correlation will drop. And you can do this analysis in the reverse order, where you can build a template with post-stimulation data, correlate this trial by trial, and now in this case, these correlations will be low, and then when the cell remaps, the correlations to the post-stimulation template will be high. And the point in which these two switch is showing the trials in which the cells are remapping, and the difference between these two lines is giving us some information about how distinct are the pre- and post-stimulation templates in driving the trial-by-trial activity. And so in our CA1 stimulated case, you can see that around the time of our stimulation, we get these mismatches between the templates, which gives our metric for the remapping of the cells. But we see the same kind of thing also on the non-stimulated shanks. And both of this was more than in control sessions where we didn't actually apply any optogenetic depolarization of the pyramidal cells. And I should say that the opsin is expressed in the CAM kinase positive cells. So what was very striking to us is we reproduced these experiments in other animals in CA3 in the dentate gyrus. And now in both of these regions, our depolarization was actually not sufficient to induce place field remapping. And typically, these regions are thought to be the most plastic areas of the hippocampus, very important for one-trial learning, but at least with this artificial stimulation, it looks like it was not able to induce long-term plasticity in the hippocampal place fields. And so as I mentioned, the place fields could actually remap to anywhere on the track. So we wanted to get a handle of, can we predict anything about the dynamics of this remapping? So what I'm showing you here is for all of the cells that remapped in CA1, I align the activity and the position to the place of the future place field, and I align all this activity to the time of the first stimulation. And so you can see that these place fields seem to ease in here, but even before the stimulation, you can see these sporadic spikes in the location where the place field will be, as though there was some subthreshold drive in that location. No one would call it a receptive field, but it's some hint that these neurons are already primed to fire in this location. We wanted to look at this a little bit deeper, and so we wanted to see maybe there's some hints of this kind of structural constraints to where the place fields can remap that we can get at the activity during sleep even before we put the animal on the track. And so what we did is we turned to analysis that was developed by Ken Harris when he was working in György Buzsáki's lab, where you try to predict the firing, the spike train of one target neuron using the activity of an ensemble of peer neurons. And so you look to see which neurons are firing at what rate. You take a weighted sum of those, and you pass that through Poisson to try to predict the instantaneous spikes of your target neuron. And so we can take data just during the sleep before we put the animal on the track, and we can look to see how these weights relate to the distance between the place fields. And when we look to see how these weights relate to the distance between the place fields before we stimulate, we can see a nice parametric relationship, where those neurons that will have place fields close together on a familiar track have these higher peer prediction weights. And so this is a nice way to look at the phenomena of preplay, though this is a familiar environment. But what about in the remapping case? So now we look at the subset of our neurons that remapped, and we look to see how is it that the correlations between those neurons and other neurons simultaneously recorded in ensemble related to the place field separation. And we saw the same kind of pattern. So if we look to see the difference between where the place fields are located after we stimulate, if I remap and my place field now overlaps with another neuron, those two neurons are going to have a higher correlation, at least as expressed by this peer prediction beta weight, as compared to two neurons that have non-overlapping place fields. So this suggests that quite a lot of the dynamics and the structure that's already intrinsic to hippocampal area CA1 is dictating how these neurons are allowed to remap in this environment. And so what we predicted is maybe what's going on with this remapping is it's actually some subthreshold drive to those locations. And with our optogenetic stimulation, we're allowing those subthreshold place fields to be disinhibited. And so in a previous set of work, what we did is we looked to try to estimate what is the excitatory drive to local inhibitory cells, looking at these kind of cross-correlation shapes. And so since the 1960s, people have been looking at the cross-correlation between pairs of neurons and seeing these sharp peaks, as you might expect if you observe a real synaptic connection between them. And in a set of experiments, we actually drove a presynaptic pyramidal cell just juxtacellularly, and were able to observe postsynaptic spikes in an extracellularly recorded interneuron, confirming that indeed, when you see these kinds of sharp peaks in your cross-correlation, it's indicative of the existence of a synapse. And you can build these kinds of connectivity maps, functional connectivity maps, where you can, in this case, it's the connections between all of the pyramidal cells that we're recording and simultaneously recorded interneurons shown as circles. And we can color code these connections in terms of what's the reliability of these spike-spike couplings. But this is a static image. What we want to do is we want to see, do these functional coupling strengths change over time? And particularly, do they change with the stimulation? And so we built a generalized linear model to try to predict what is the influence of our presynaptic pyramidal cell in driving the firing of a postsynaptic interneuron, regressing out slow fluctuations in the postsynaptic interneuron firing rate. Now we allow that coupling strength to change over time, over a slow time constant over the course of minutes. And what we can see is that actually the coupling strength between the pyramidal cell and the interneuron is quite dynamic over the course of hours, which you can see in these cross correlations here as the size of the peak above the baseline. Of course, we're recording more than just one pair of these cells. When you record an ensemble of these synapses, pyramidal to interneuron synapses, you can see that they're highly dynamic, but it also looks like there's subnetworks here, where certain pairs of synapses tend to be very effective at the same moments at the expense of other pairs. And then these modes seem to switch. And so if I want to describe all of the dynamics in this kind of matrix here, I obviously don't need to consider all 60 of the synapses. It looks like there's some latent low-dimensional structure with which I can describe this dynamics. And so what we did is we performed a principal component analysis and independent component analysis to try to get at a handle of what is this latent structure here. And in particular, what we did is we detected the independent components prior to stimulation. We looked to see how strongly those were expressed post-stimulation, to give an idea of how is it the pairs of synapses are changing in their co-fluctuations around the time of our stimulation. And we can do the same analysis now, trying to detect the independent components after stimulation and look to see how strongly they're expressed beforehand. And what we observed is that with the stimulation, those independent components are not so strongly expressed before and after stimulation, suggesting a reorganization of which of those synapses are allowed to fluctuate with one another, as compared to our CA1 control cases, where we did not observe remapping, and also compared to CA3 and the dentate gyrus, in which those place cells did not remap. So in conclusion, it looks like our focal optogenetic stimulation induced place field remapping in CA1, but less so in CA3 and the dentate. And this also correlated with changes in the excitatory inhibitory coupling in the CA1. For me, the implications are that maybe these changes in lateral inhibition could unleash some pre-existing state. So when I think about learning, I think about it as a hypothesis test. I think the world works in this particular way, so let's try it out, and maybe it doesn't, and we want to clamp down a whole different correlation structure and try something new. And I see that the interneurons could be a very effective locus for that kind of plasticity. And in particular, this means that it could be an unappreciated role for memory storage in areas without strong excitatory connections, like in CA1. And I hope that you appreciated how dynamic that synaptic coupling strength was. And to me, this suggests maybe actually the synaptic strength is being modulated by the ongoing activity in the network, rather than some slowly changing entity over which fast action potential activity flows across. So with this, I'd like to say my acknowledgments. I'm a postdoc in György Buzsáki's lab. He's been a fantastic mentor. Roman Huszar did the statistical modeling of the synaptic strength dynamics, and Dan English did the synapse detection work with me. Kanghwan Kim and Euisik Yoon developed the micro-LED probes at the University of Michigan. And I've actually just signed a contract to start a lab at the University of New Mexico, so if you're graduating soon and you'd like to work on these kinds of questions, please be in touch. Thank you.
I was wondering whether there's any regularity in the position of these subthreshold fields relative to the original field.
We don't know. I don't know. So if I can't predict where the place field will remap to as a function of where the existing fields are.
Okay.
Okay. Thank you.
Thank you.
Should I start?
The last speaker for this session is Pinglei Bao from Caltech.
So we have in the previous talk about how the brain computes where we are. An important part of knowing where we are is by recognizing the object around us. So for example, some of you might recognize a cal tag from this canoe. In my talk, I would like to talk to you about the object recognition in the primate brains. Recognizing visual object is critical to survival. Mistakes in object recognition can even be fatal. For example, fail to recognize there's a snake here or a white track here. Many high-level processes in the brain act on objects. Memory, attention, decision-making, and social cognition all depend on object recognition. So how is object recognition actually accomplished by the brain? Here's a side view of the macaque brain. Object recognition happens in this region here, IT cortex. Here's a rough picture of our current understanding of how IT cortex is organized. Several regions within the IT cortex have been identified as being specialized for specific category or stimulus dimensions. But there's a very large region of IT cortex that is completed uncharted. How do we go about understanding these uncharted regions? To address this, we began with anatomy. Specifically, we first run a large series of localizers to identify all the islands that we have already been mapped in the IT. And then we insert the electrode purposefully into this unmapped territory. We electrical microstimulate a piece of this unmapped territory while performing simultaneous fMRI to ask what other parts of brain light up. Stimulation of this region revealed that it's connected to other discrete regions in IT cortex, one more posterior and one more anterior. And here you can see the fMRI response time course from these three regions. The bump in the red traces corresponds to the block of electrical stimulation. Thus, we have discovered a new anatomical network of connected patches in IT, where we call the network X for the time being. To understand the function of this network, we present a stimulus set consisting of 51 objects, and each at 24 different views. Here, the response of cell in the 3 patch of network X to this 51 object. Each row represents 1 cell, and each column 1 object. The cells are grouped separately for the 3 patch. You can see quite a high consistency in the selectivity of cell within the network. All the cells in X1, X2, X3 are separated by the faces, and many show a strong response to a few of the objects shown here. These objects are distributed across different categories. So we have found a new functional network in IT whose cells show the consistent selectivity, but network doesn't seem specialized for a specific category. Does this network exist across animals? We next performed an fMRI experiment where we present those most and least preferred objects determined from the cell recording the 3 patch in our first monkey. And here's the result of the stimulus comparison in the fMRI experiment in the first monkey. You can see the exactly overlap with the patch of network X revealed by the microstimulation. And in the second animal, when we performed the stimulus comparison, we again saw a network consisting of multiple patches in approximately similar location as the first animal. Here, the response of cell recorded from the 3 patch in the second animal, you can see they are highly consistent with each other and with those in the first animal. So we have identified a new object network in IT cortex that exists across animals. And one of the hallmarks of the face patch is increasing viewing variance as a go anterior. We examined the viewing variance of cells in the different patch of network X and found a striking difference. Cells in the most posterior patch of network X were view specific, while those in the most anterior patch were viewing variant, just like in the face patch system. The viewing variance in the most anterior patch of network X is very surprising, since the monkey had no experience with many of these objects, for example, this airplane. So this must have learned through the experience with other objects. What is a network actually coding? Here again, the top and the bottom 5 stimuli for this network. We notice that all the preferred stimuli contain the same protrusions. To further test this, we create a simplified stimulus set consisting of light segments varying curvature, aspect ratio, and orientation. We record neurons from X2 and X3, and these 4 groups correspond to the 4 different aspect ratio levels. The neurons show a very strong, consistent preference to a high aspect ratio in both X2 and X3. So one feature cells in network X care about strongly is the aspect ratio. But is there a systematic way to understand what the network X is coding that doesn't involve guessing? The key challenge here is to develop a way to parameterize the object. In other words, to describe an arbitrary object with a small set of numbers. Until a few years ago, this still was an impossible problem. But now, with deep network trained on object classification, we have a great solution. Deep network provide a very powerful way to parameterize the arbitrary object. In the sense, for every object, you can get out a few thousand numbers describing the object at a high level, which can be used to classify the object with a linear classifier. And then we even can use PCA to further reduce the dimensionality of the object space. So now we can describe each object by 15 numbers. What do the resulting object axes look like? Here you can see the object within the highest and the lowest projection on the first PC, computing using activation of units in AlexNet layer FC6. It seems to correspond to the spiky and the smooth things. And here's the second PC. Clearly, there's a difference in terms of curvature between the 2 groups of stimuli, but just for simplicity, I'm going to call the two ends of the second axis as animated versus inanimated. So we now have a set of objects axes spanning the object space. When we analyze the response of cell in the network X to object parameterized by this object axis extract from the deep network, we found single cells show the ramp shape tuning to the subsets of objects. For example, these cells show a minimum at this extreme and a maximum at this extreme of PC1. So we have discovered a new network in IT cortex. Then this network is composed of the connected patches. The patch has a consistent selectivity, and there's a increasing viewing variance from the posterior to anterior, and the single cells use the axis code. But network X covers just a small portion of the unmapped territory of IT cortex, as there are other similar organized networks in IT. And the more important question is there a general principle governing how all of this network organized? If you have been paying attention, you might have noticed a very weird coincidence. Here are the 5 most and least preferred objects in network X, and here the highest and lowest projecting image onto the PC1 of AlexNet. Do you notice a resemblance here? This is extremely weird because network X was just a network that we discovered by chance, by stimulating a random piece in IT. How can selectivity be so perfectly aligned with the first PC of AlexNet, which knows nothing about the IT cortex? We thought about this very hard. Then we realized it might not be a coincidence. Recall the first 2 axes of object space. Faces are the stubby animated objects. Bodies are spiky animated objects. Network X seem to prefer the spiky objects regardless of animacy. This observation made us wonder if it's possible that IT might be topographically organized according to the first 2 dimensions of the object space, in the same way that the retinal topic is organized according to the polar angle and eccentricity. If this were the case, then the preference of network X for the spiky object is no longer such coincidence, because we are proposing that stubby and spiky is a basic organization axis for the IT. As a first step to test this, we project all the stimuli that we shown to the monkey onto the first 2 dimensions of the object space. And then we marked top 100 images for network X, the body network, and the face network. You can see that they approximately span the 3 quadrants of the space, but there's a region of the space that's not represented by any of the cells. If it's true that IT is laid out according to the general scheme shown here using the first 2 dimensions of object space, then we predict that there should be a 4th network representing objects projecting strongly onto this region, namely stubby inanimated objects lacking protrusions, like a USB sticker or radio. To test this prediction, we first run an fMRI experiment with 4 different blocks corresponding to the 4 quadrants of the object space. When we compare the stubby versus other blocks, we found a network consisting of multiple patches selective for the stubby object, shown here on the coronal and sagittal slices. And here's the electrode entering the anterior stubby patch. And here the response of 2 cells we recorded from this patch. You can see both cells prefer the stubby object, like the truck and these houses. Here's a population plot with the 5 most preferred and least preferred objects shown below. You can see it shows a very different pattern from the network X and the body patch network. So the hypothesis that IT is organized according to the first 2 dimensions of object space has actually let us discover a new shape network. When we project the preferred object of stubby patch cell onto the object space, indeed, they most align the previous unrepresented region of object space. Here you can see the 4 network, body, stubby, body, face, stubby, and spiky overlay on the coronal slice. You can see a progression from the body to the face, to the stubby, to the spiky in both hemispheres. The spatial organized is preserved in the middle and anterior nodes of this network: green, blue, purple, orange. Green, blue, purple, orange. And the organization holds true across animals. To summarize, here's a current big picture of the spatial organization of IT cortex for representing the object shape. IT contains at least 3 distinct mappings of object space, with increasing viewing variance from posterior to anterior, And each mapping is further split into at least 4 regions obtaining the 4 quadrants of object space. The sequence goes from the body to face to stubby to network X. In this picture, the face patch are not special. For previous people have thought that the face patch system arose due to the social importance of the faces. In this new picture, we believe the face patch system arises naturally from the statistical structure of object space. And importantly, a large swath of IT cortex follow the same principle with similar modularities. So going back to our original picture, how many islands do we need to fill all of IT? So far, I have shown you that 4 networks can build a complete object space. If we add up the area occupied by these 4 networks, together with color, disparity, and the scene network, they can currently explain about 50% of IT. There's clearly some work to be done. Additional network related to the 3D object and motion might be found in ID cortex, since this information can be found in the natural stimuli. But this dimension won't include it in any of our current stimulus set. But I want to show you a last piece of data that suggests even just these 4 networks already carry a fairly complete code for the object. The way we access is by asking how complete what is, the way we assess how complete the code was to ask how well can we reconstruct an arbitrary object using activity from just these 4 networks. To address this, we decided to try to reconstruct an arbitrary object from the responses if cells from these 4 networks and see how well we can do. For example, if we present a giraffe and record activities of neurons from the 4 networks and try to reconstruct the giraffe from this activity, would bear some resemblance, or would the result be so impoverished because these 4 networks are such a small part of the full decoding scheme of our IT that the result would be unrecognizable. Since cells in the 4 networks show a ramp-shaped tuning to a subset object feature, this means we can write the response of each cell as a linear combination of the object feature, so that we can predict the object feature also from the population response of vectors by a linear transform. To reconstruct the object, we use a generative adversary network that was trained to invert AlexNet. AlexNet converts the image into the compressed feature vector, and again can take the compressed feature vector and turn them back into the images. We use response of IT cell from the 4 network to predict the feature vector corresponding to an object, and then run it through again to reconstruct images. And here are some example instructions. Well, they are not certainly not perfect. They confirm the richness of the object code containing just a few hundred neurons within the 4 IT network. To summarize our current picture of the IT organization, we found the cells anatomical class into the 4 networks according to the first 2 components of their preferred axis, forming a mapping of object space. This mapping repeated at least 3 times with increasing view invariance. And the cells in this mapping have sufficient coding capacity to reconstruct general objects. So I think what's really exciting here is that we have changed the basic understanding of the ID cortex. It's not a homogeneous land. It is also not an ocean with several islands. Instead, it contains an organized map like many other brain areas. And discovering this map is important because it shows that IT organization is governed by a general principle. It gives a handle to uncoded cortex, IT cortex. And finally, it seems like the downstream area are aware of this map and influence how objects are read out in the later stage. So thank you thank you for my collaborators.
Thanks for the great talk. I think this is really cool. So my question is, like, we know objects live in very high dimensional space, and like, there's, I guess, nothing really particular about 2 dimensions. So my question is, if you, for example, look at like principal components more than 2 dimensions, do you still see these kind of like smooth tiling through like a higher dimensional object space?
We look into we cluster neurons based on the only first 2 dimensions, or based on the later axis, later dimensions. We only found the neuron cluster in the first 2 dimensions. We don't find the cluster in the later in the other dimensions. But the thing might be changed if we record it. We have more dense recording. within a very small range, something might not be, these things might not be, I'm not 100% sure when the cluster cannot be seen in a small range, but it's possible. But so far, we don't have evidence for the cluster in the other axis.
Thank you.
Looking in the AlexNet at these principal components in terms of how they affect categorization, did you come up with any hypotheses for sort of normative explanations for what the point of, say, a stubby dimension is?
Sorry, I'm not clear. Can you repeat your question again?
So it seems like you found in AlexNet that sort of spiky objects, for example, in these different categories that you came were sort of a big part of population representation. Did you look into AlexNet and see if there was any indication of what function these were sort of performing in categorization? Is it that many categories are separated by whether they are spiky or stubby, or is it a more distributed coded that later gets decoded into?
I haven't looked into that, but that's a very good question. It basically you're asking what's the function role of these different axes. I think if you do PCA for PC1, do account for larger variance of these units. It must play a very important role in the later stage of the object recognition for the classification. I haven't looked into that, but I believe there must be. Otherwise, why they have the PC1? Why the PC1 is stubby but spiky, right?
Sure, very interesting to see. Thank you.
Yeah.
Thanks for your fantastic talk. I'm wondering whether there is some symmetric responses of neurons with respect to the symmetry of stimulus on the circle spanned by the PC1 and PC2 in your object space.
Sorry, can you repeat your question?
So could you go back to your slide of a map of object space? I mean, you arrange the stimulus on a circle, and the circle is, go back.
Okay, I mean.
Yeah, this figure. So I mean, so the stimulus are arranged on a circle.
Yes.
So if a stimulus in the lower left part, the blue region, and we could also, and for another stimulus in the orange region, they are symmetric of the ranging on this circle.
Yes.
So I'm wondering whether you have found some neurons who show symmetric response to the pair of stimulus in the blue and orange region.
Yeah, interesting. I never found a neuron who is also prefer the face, also prefer this spiky object. Also, if you look at my spiky neuron recording, I never found one single neuron also shows a high response to faces, also to the spiky object. I think maybe that's a very strong evidence showing that this IT cortex map is really coding with this large PC of the object space.
Okay, thanks.
Last question, please.
Very interesting work. It struck me that it's particularly interesting how one of the principal axes is animate versus inanimate. And as we live in an increasingly urban world, I wonder if you were to take monkeys raised in environments surrounded by USB sticks compared to giraffes, you would see an overrepresentation of those inanimate objects.
So I think the words I use, animate and versus inanimate, it's not a semantic category. It's more like it's not just a simple way to describe the two types of stimuli. I think these two types of stimuli, the main difference is still about shape. It's a curvature shape. Do they contain the curvature or not? That's what I don't think semantic meaning plays an important role here.
Sure. But in classic visual tasks, raising cats in environments with certain orientation can bias visual cortex. I wonder if this would progress higher up into the visual hierarchy.
Yeah, maybe, yes. Yeah. Yeah.
Thank you.
Thank you.
Okay, let's thank all the speakers of the session.
Session entitled What and Where Is That Smell? Our first speaker today is Rainer Friedrich, and he's going to talk about connectivity and computation in olfaction.
Hello. First of all, I would like to extend a massive thanks to the organizers for inviting me here. For about a century, more than a century, we had very detailed anatomical data, yet it was not possible to actually reconstruct wiring diagrams of neuronal circuits, mainly for two reasons. Because these data that Cajal and Golgi, for example, allowed them to get, missed two important parts here. One is that in order to resolve the morphology of these neurons in great detail, they had to be stained sparsely, so a dense reconstruction of a circuit was impossible. And also the light microscopical techniques to draw these neurons did not allow to resolve synapses and to decide which neuron actually makes a physical contact to which other neuron. So this has changed recently with the advent of various techniques, in particular three-dimensional volumetric electron microscopy techniques. And that now puts us in a position, at least in theory, to obtain detailed wiring diagrams of relatively large neuronal circuits. So before we go there, maybe let's just think a bit of why would we like to do this. So of course, this speeds up anatomical projects quite a lot. So we get very detailed information about morphology, anatomical cell types, from which we can then derive interaction probabilities. What's the probability of cell type A to make synapse onto cell type B? This sort of thing. So I would call this high throughput anatomy, and this alone is actually a motivation for many people to go to these dense three-dimensional reconstructions. However, what I'm more interested in is actually questions that relate to the higher order structure of neuronal circuits. So, for example, we have so far been unable to exhaustively analyze what is often called circuit motifs. So we can, for example, do experiments using, let's say, paired recordings or rabies, transsynaptic rabies tracing to identify neurons that make synaptic inputs onto a single cell. But then if we want to ask the next level question, whether perhaps these presynaptic neurons are more or less likely than average to make connections amongst themselves, this is what essentially all techniques available have not been able to do so far, except for dense circuit reconstructions. And of course, when you then extend this and ask, can we reconstruct the connectivity of connected ensembles of neurons, this is where dense reconstruction becomes essential. And obviously for networks like these ones, if we want to understand those, connectivity is of a major importance. So here you could argue that in this visual pathway, neurons of the same layer may actually be identical, and their actually individual identity is imposed upon them by connectivity. So what distinguishes one pyramidal cell from another may be actually the inputs and outputs of those cells. So in order to understand the functional organization of such circuits, we need to actually resolve the dense connectivity of the network. And these notions, of course, also apply to artificial neuronal networks. Why don't we just simulate those? I'll just give you a little number game here. When you have 3 neurons, the number of possible configurations by which you can connect these 3 neurons is 81. If you have 17 neurons, that number is greater than 10 to the 86. And 10 to the 86 is a meaningful number because that's the number of atoms in our universe. So most brains have substantially more than 17 neurons, which means that it's very hard to actually just simulate all the possible configurations. So this is why people like us decided we want to actually measure the wiring diagrams. And we then want to relate the wiring diagrams to functions of neuronal circuits. We do this in the olfactory system. The olfactory cortex is believed to be an autoassociative memory area that also represents objects. This is part of what we work on, but I will actually have only one slide here about this because also the following talks, 2 of them will actually go into this direction. So we work on zebrafish, And some recent work from our lab showed that the zebrafish homologue of olfactory cortex and telencephalic area called DP actually contains ensembles of neurons that are characterized by their correlated activity. These ensembles become more prominent when you train an animal in an odor discrimination task, and in particular they represent the rewarded stimulus more than before. In another study, what we saw is that the excitatory and inhibitory inputs to individual excitatory neurons in this cortical area are actually co-tuned with respect to different stimuli. And this is something that you would expect actually from an auto-associative memory network, because as you increase some connections, form ensembles, you would increase the gain among these neurons, and this has to be counterbalanced by an increase also in inhibitory gain to prevent runaway excitation. So this leads to actually a strong prediction that excitation inhibition should be co-tuned, should become co-tuned when the network learns something, and this is what we actually observed experimentally. And we also found that inhibition actually contributes to the specificity of responses over here, and that this inhibition is actually modified during learning. Okay, so these observations kind of, for us, strengthened the notion that the olfactory cortex is some form of memory network, where connectivity is changing as a function of experience. Now, today I will, however, focus on the olfactory bulb. Now, if this is a memory network, then the olfactory bulb should, of course, do some pre-processing that allows the memory network to store the relevant information. So, therefore, this olfactory bulb, which is obviously a sensory circuit, should, for example, filter out features from the sensory input that are relevant, should discard other features, and should decorrelate odor-evoked activity patterns in order to allow for more efficient storage in this memory network. And that's actually what we find the olfactory bulb does. So we therefore think that, in a sense, both of these circuits are memory circuits. This one here learns during experience, so during the lifetime of an individual. Now this one here has learned something about the stimulus space over the course of evolution, because in order to filter out relevant features, the network has to learn something about the statistical structure of the olfactory stimulus space in order to then maybe select important features and suppress less important features. Okay, how does the olfactory bulb work? Here's a brief overview. We have these olfactory glomeruli, and as you all know, a glomerulus receives input from sensory neurons that express the same odorant receptor in the nose. These sensory neurons then make synapses onto mitral cells and onto interneurons, and these mitral cells are the output neurons of the olfactory bulb. They do not make direct excitatory connections amongst each other, but they communicate via at least two synapses via these interneurons, which are mostly or exclusively inhibitory. So an odor would then activate a particular combination of odorant receptors, and therefore a particular combination of glomeruli, as shown here, and that evokes an output from the olfactory bulb. Now, when you use chemically similar odors, you will get overlapping patterns of activity across this array of glomeruli, and this is probably an unavoidable consequence from the fact that there are, what did I do? How can I turn this off? I have no subtitles. I'm just improvising here. Let me. Okay. Does anybody know how to turn this off? I think I had some. Okay, great. Thanks. Whatever you did. Okay. So these overlapping patterns of activity then become transformed by the circuitry in the olfactory bulb, and as a consequence of this, there are a couple of normalization operations that occur, and overlapping patterns actually become decorrelated. So together, these transformations can be described as a form of whitening, because similar odorants, characterized usually by shared functional groups here, are sort of clustered in the representation space of the glomeruli, but then they become declustered, meaning decorrelated and variance normalized in this output space. So in the first approximation, this transformation can actually be described as a whitening operation. So here in a little more detail, this is just examples. This is glomerular activity in the zebrafish olfactory bulb evoked by a particular stimulus, and these are now activity measurements from the output neurons, the mitral cells, in response to this odor. What you can see is that there's initially some dynamic phase, and then it approaches a steady state. These are responses to a second similar odor, and when you now look at the early activity of the output neurons, you can see by eye that this is highly overlapping, just like the input activity is highly overlapping. But when you look a few hundred milliseconds later in time, these patterns become less correlated. And when we just measure the correlation coefficient as a function of time, you see that it decreases. And when you repeat this experiment for all the inputs that give highly correlated, all the stimuli that give highly correlated inputs, you see the outputs are also initially highly correlated, but then these correlations decrease. So we have a pattern decorrelation. This is the correlation matrix as a function of time. You find that there are groups of stimuli whose activity patterns are initially correlated, then decrease, and these are groups sharing similar functional groups. Okay. So we have this pattern decorrelation. Perhaps one word about the time scale. This takes a few hundred milliseconds to occur here. This is probably due to the fact that it takes us a few hundred milliseconds to ramp up our stimulus, so there's a technical limitation. When we stimulate optogenetically, this can be a lot faster. And the group of Emre has recently actually looked at the flicker fusion frequency of sensory neurons in the nasal epithelium of a zebrafish larva, and finds that this is on the timescale of an interstimulus interval of about 10 seconds. So we believe that essentially for the purpose of odor processing in a zebrafish, which seems to be very slow, this is almost near instantaneous decorrelation. Okay, so does it correlate to behavior? And indeed, the lab of Tine Valentinjic has measured the ability of zebrafish to discriminate between all of these odor pairs, and they found only one pair that the fish couldn't discriminate, and that actually happens to be exactly the pair where the representations don't decorrelate. So it seems to be valid for behavior. And Alan Carlton's lab has, in mice, addressed this question more exhaustively, and they also find a very strong correlation between the decorrelation of odor pairs in the olfactory bulb, which actually in a mouse occurs during a single sniff, and the ability of the mouse to learn that discrimination. Okay, now how does decorrelation work? Let's look at some more data in more detail again. So what we see here are responses of 50 or so mitral cells to 16 or so odor stimuli averaged over trials and everything. And the neurons have been sorted by k-means clustering in such a way to bring out some clusters that we observe occasionally. And in this instance here, we found the clustering reveals 4 odors that evoke similar patterns of activity. What was this? Ah, now I found it. Similar patterns of activity. And these patterns of activity are actually similar because there's a subset of mitral cells that respond strongly to those stimuli. Now, how can this be decorrelated, these activity patterns? So for a long time, there has been a notion in the field that this has to do with contrast enhancement. This idea has been probably borrowed from the retina. There have been different mechanisms proposed how contrast enhancement could occur in the olfactory bulb, but essentially what they all do is they decrease activity of weakly active and increase activity of highly active cells. That's obvious. However, if you apply this transformation using Photoshop or something like that, obviously you see that you don't decorrelate these patterns. So the statistical structure of these activity patterns in the bulb is actually such that a contrast enhancement will not result in a decorrelation, at least in the zebrafish. So how can it then work? Now you can say, okay, there are these highly active cells. What if I would just selectively decrease, again using Photoshop here, the activity of these cells? then it looks actually more decorrelated. And then we can look just in the decorrelated state a few hundred milliseconds later what happens, and that is actually not such a bad approximation of what really happens in the olfactory bulb. So there seems to be a transformation such that these highly active cells actually their activity becomes suppressed, while the activity of other cells, other odor responses, is not affected very much. Now this means this is a seemingly simple change in activity patterns, but it is something that cannot be explained by a global transformation, such a normalization or something like this, because the activity of these cells should be reduced only if one of these 4 odors is present, or if the other cells are active at the same time. When it's one of these odors here, then the activity should actually not be suppressed because they are informative about those odors. And likewise, when one of these 4 odors is present, then only the activity of these cells should be suppressed and not the activity of other cells in the circuit. So in order to achieve this, you can quickly arrive at the conclusion that you need some kind of connectivity that is not random connectivity, that is not global connectivity, but that is somehow must be adjusted to the tuning curves of these neurons. So how does that work? And in order to get there, we need to reconstruct the wiring diagram. So the main questions that I will address in the rest of the talk are therefore about the mechanism of whitening. Can we actually explain this mechanism if we know the wiring diagram? But this will also actually have some broader implications, because for a computation like this, we didn't have a precedence where we could go to the literature and ask how much information about a computation like this can we actually obtain if we would know a wiring diagram, because for similar computations, wiring diagrams have not been available. So there have been debates in the field how much information is actually in the wiring diagram, so we would like to address those. And rephrasing this question or a similar question from an experimentalist point of view would be, do the techniques that we have available today, the electron microscopy, reconstruction methods, which are not perfect, but are they already sufficiently good to extract enough information from the wiring diagram to explain anything useful? So what we used as an approach was zero block phase electron microscopy that was pioneered by Winfried Denk, where we embed the sample, put it in a scanning electron microscope, take an image from the block phase, and then an automated ultramicrotome will automatically, in the vacuum chamber, shave off at 25 nanometer sections. The next image will be taken, etc. And this can give us then images that are already quite well aligned. So here's an example using modern techniques. You can, by eye, obviously appreciate that it's now not a problem for a human, at least, to follow processes here, identify synaptic vesicles, synaptic contacts, etc. So we did this a few years ago when this was still quite an effort, but still a graduate student in the lab, Adrian Wanner, managed to recruit lots of external helpers and with them actually reconstruct all the neurons in the olfactory bulb of a zebrafish larva, which are just about 1,000 neurons. And we can then, of course, do high throughput anatomy. We can classify these neurons based on morphological features, assign them to glomeruli, map these neurons in quite some detail. This alone was actually very informative. So we found that a major population of interneurons, the so-called granule cells, was not present yet at this early developmental stage. We identified some rare cell types, quite nice. But what we were really interested in then was to take the next step to synaptically connect these neurons. So again, manually, we annotated all the synapses. And just as a reminder, we found many of these so-called reciprocal synapses within the olfactory bulb. You probably know that many neurons in the olfactory bulb have neurites that are actually both pre- and post-synaptic to other neurites. So they can therefore both make and receive synapses. And often you have these pairs of reciprocal synapses between a mitral cell and an interneuron, where an inhibitory synapse is actually paired with an excitatory synapse on the same dendrite. And in fact, we found that about 50% of all the synapses in the olfactory bulb are part of these reciprocal pairs. So with this, we then arrived at this, which is the wiring diagram, where we have mitral cells that are not connected to each other, and then interneurons that are connected to mitral cells and also amongst each other. So can this wiring diagram now explain something about the function of the olfactory bulb? To do this, we actually, before we prepared the sample for the electron microscope, we also measured odor responses of about 30% of all the neurons in the same animal. And then we can merge the data sets to identify the odor response, to map the odor responses onto the wiring diagram. So the activity data set then looks like this. We had 8 different stimuli, got odor responses from mitral cells and interneurons. Then we temporally deconvolved the calcium signals to estimate the underlying firing rate changes. And then to simplify analysis, we just picked 2 time windows, one early one that should correspond to the correlated state, and a later one, about 1.6 seconds later, that should correlate to the decorrelated state. And we picked those such that the mean activity in these 2 time windows is not significantly different. So anything you see cannot be explained by a simple change in mean activity. This was a zebrafish larva. Previous results were obtained in adult fish. Do we still see a decorrelation? And in fact, we find that patterns still become decorrelated, despite the fact that there are a lot fewer interneurons. This is now focusing on the bile acids, where the correlations were initially particularly high. And you can see the correlations go up, and then they decrease, even though the activity is not significantly different between these 2 time windows. So we have a pattern decorrelation as we observed it before. Now, what can we explain about the pattern decorrelation knowing the wiring diagram? So in order to address that, we made a network model, a simulation, where we modeled the individual neurons using what we thought is the minimally complex model of a single neuron, which is just a threshold linear firing rate unit. And then we connected these units up using the wiring diagram that we found. The synaptic strength of all neurons of the same class are the same, no matter where they land on. So we have actually a very small number of free parameters in this network. So essentially, by putting in the wiring diagram, we reduce the number of degrees of freedom of something near 1 million to 6. So does it still reproduce what we saw experimentally? This is the activity, time course of the activity, and now importantly, this is the time course of the correlation, so it indeed goes up and decorrelates. We then asked what the function of different types of connections is, and in particular, we reduced the strength of these interneuron to interneuron connections that reduced activity consistent with a disinhibition that is mediated by these connections, but it basically had no effect on decorrelation. So for any future analysis, I will therefore omit the interneuron-interneuron connections and focus on the mechanism of decorrelation. So what we can now obviously do is randomize the wiring diagram and see what we get then in the simulation. And it has very little effect on the mean activity, but decorrelation is basically completely abolished. This is the amount of decorrelation between the 2 time points observed experimentally in the simulation and with a randomized wiring diagram. We can now do more sophisticated manipulations and randomize or shuffle the connections, feed-forward connections and feedback connections using the same shufflings. So what this will result in is a shuffling of the disynaptic connectivity. So from mitral cells disynaptically to other mitral cells, it will shuffle this connectivity without changing the statistics of these disynaptic connections. And that actually also completely abolishes the decorrelation. So that means when we manipulate who is disynaptically talking to whom, we affect the decorrelation. That means the connectivity is adjusted to the input, to the structure of the inputs here. So therefore, when we shuffle the inputs, we should also lose decorrelation, and that's true. Okay, so how are these disynaptic connections organized? To go there, we analyzed these potential motifs in some more detail. And we classified the motifs in 4 different classes depending on the number of disynaptic reciprocal connections that are contained in these motifs. So here we have 1 reciprocal connection, here we have 1 reciprocal connection in another direction, and here we have 2 reciprocal connections. And then we find that motifs with reciprocal connections, in particular this one, are strongly overrepresented against random controls. Now we can ask, if we have triplets like these, is there any relationship in the tuning curves of the mitral cells that are part of these triplets? And when we analyze this, we actually find a relationship like this. So this is showing that when the mitral cells are more similarly tuned, they are also more likely to interact disynaptically via interneurons. So this is actually the kind of curve that I wanted, working on the olfactory system, that I wanted to have for many, many years, because this tells us how lateral inhibition, if you want, is organized in the olfactory bulb with respect to tuning, which is something that was impossible to analyze because it requires tracing across 2 synapses and not just 1 synapse. So paired recordings or so won't do it. If we now include only reciprocal connectivity, we find basically the same thing. So that holds, it's actually even more pronounced if we look only at reciprocal connectivity. So now you may say, aha, I've seen this before. This is common in the visual system, in the retina, for example, where neurons with similar tuning do lateral inhibition onto each other, and that sharpens tuning. So this is, however, not the same as in the olfactory bulb, and it's quite obvious why. So for classical sharpening, contrast enhancement type of computation, you would need independent synaptic pathways connecting these 2 cells, because if you have a slight asymmetry in the input, then there should be a winner-take-all effect, and therefore only 1 pathway can be active and the other one should be off. However, what we have in the olfactory bulb are these reciprocal connections. So that means the interneuron gets the same input independent of whether the asymmetry in the input is this way or that way. So therefore, the interneuron always then provides the same inhibitory feedback independent on how the asymmetry, or if there's an asymmetry in the input, is actually organized. So therefore, such a circuit should do something that we call feature suppression. That means because if there are cells with similar tuning, they would respond to something that can be called a feature. And if there's now a cohort of these cells activated by this feature that connect to the same interneuron, the interneuron will then provide strong reciprocal inhibition to this cohort and suppress the representation of this feature, usually providing more inhibition to this cohort of neurons than to the average population. So therefore, a reciprocally connected circuit like this will perform feature suppression rather than contrast enhancement. And this is actually exactly what you would like to suppress the activity of these neurons. If you would connect up these neurons in such a feature suppression circuit, then whenever one of these stimuli is active, it should activate these neurons together relatively strongly, and they should then downregulate their own activity. So we can now we have a hypothesis that we can test using our wiring diagram in more detail. So again, the hypothesis is that we have neurons that get activated and respond strongly to similar odors, and these neurons are connected into cohorts via these disynaptic reciprocal connections to interneurons, and they would then downregulate the activity of the neurons within the cohort, but not affect the activity outside the cohort very much. And this should lead to a pattern decorrelation. So can we actually see this in our results? So I'll walk you through this, through an analysis that asks that question. So this is an example of responses of mitral cells to 2 different stimuli, and the activity patterns of these evoked are by these stimuli early on are similar. This is the correlation coefficient. And what we've done here is we sorted these mitral cells by their individual contributions to this correlation coefficient. Those that drive the correlation coefficient up are high here. And the color now encodes for their activity. So these cells are actually also highly active. So this assumption that we've actually seen before, that the cells that are responsible for high correlations are also highly active, is actually true. And now, when we look later in time, we find that the mean activity across this population of neurons has not changed much, except for this small population up here. Their activity has been decreased, and that should result in a lower correlation, which we can actually measure. So this is an example for one odor pair. So we have done this for all the odor pairs that start out with high correlations, sorted in each case the neurons for their contribution to the correlation for that particular odor pair, average over this. And then you see that, again, still the activities of these neurons, of the highly contributing neurons, is high, and then decreased at the second time point. This is, that's just a blow up of the upper part of this curve. And the actual contributions to correlations are shown here. So the contributions to correlations are really strongly suppressed. So this is what we observed experimentally in activity measurements. Now, what about the simulation? And in our simulation, there's a one-to-one correspondence, right, between each neuron in the simulated circuit to each neuron in the that we measured from. And we find that it's exactly the same neurons where the simulation predicts that activity is reduced, and the other ones are not much affected. And therefore, their correlation to contribution drops. So that means in the simulation, we actually not just produce a decorrelation, but we recapitulate the very same mechanism of decorrelation that accounts for decorrelation in the experimental data. So that means we can now go in and analyze this more mechanistically. To do this, we can now go to the next question and ask, is the inhibition actually coming from the same cohort through this connected ensemble of neurons here? So this is just visualizing what we can do by here to trace the synaptic inputs to these neurons through two synaptic stages. So we start with neurons that are part of a cohort, excitatory neurons, the mitral cells. Usually it's about 10. Here we show only a subset of those. And then we go to the presynaptic interneurons, and then we go to the disynaptically connected mitral cells. And now if our hypothesis holds, then the neurons where we start from should be part of this ensemble of disynaptically connected mitral cells. And in fact, they are. The connectivity is relatively strong. This is now quantified here. Again, we have ranked the mitral cells according to their correlation contribution. And now we trace for each mitral cell the disynaptic inputs through the connectivity matrix. And then we find that the disynaptic input to the cells that contribute strongly to the correlation are actually coming from other neurons in that same cohort. Now you can put 2 and 2 together. These neurons are strongly active when there's a critical odor present. They also should provide strong disynaptic inhibition to each other, so they should really inhibit each other more than other neurons in the circuit. And we've seen this experimentally. Now in the simulation, we can actually also look at the synaptic input currents that these neurons receive. And in fact, they are substantially stronger than the mean synaptic input currents across the population. And we can then also randomize the connectivity matrix, and then the selective synaptic inhibition here is obliterated. So this all supports the hypothesis, but finally we can now actually do an experiment in silico that will, I think, be impossible for the rest of my lifetime as a scientist, because we can now do very precise surgical manipulations in the wiring diagram to test hypotheses. So we can, for example, now identify those cohorts and say we want to reduce the inhibition that neurons in these cohorts receive by just cutting or redistributing the feed-forward connections onto inhibitory interneurons within these cohorts. And indeed, then decorrelation is completely abolished when we cut those or when we redistribute those. And the experiment that I personally like best in silico, again, is this one. I've shown you before that when we randomize the connectivity matrix, decorrelation is lost. But if we now selectively preserve the triplet connectivity among those cohorts that we predict are important for the decorrelation of these particular odors, then actually the decorrelation is indeed preserved. So let me summarize here. We identified a microcircuit, I would call it, that produces feature suppression, where we have disynaptic reciprocal connectivity among mitral cells via interneurons. And this mediates feature suppression and was basically impossible to identify before because dense reconstructions are really necessary to observe this. So this is, in a sense, the inverse of feature amplification that can be mediated by excitatory coupling between neurons with similar tuning. And this type of connectivity has been observed, for example, by Tom Mercich-Vogel, Sonja Hofer, and colleagues in visual cortex. Now, so in vision, if we take this example, this connectivity is believed to be important to represent certain features by amplifying them. For example, to identify those objects, it could be the contours also of these kind of fish. Now, this circuit could be useful to, for example, suppress or attenuate the representations of features that are not particularly relevant for something. And this is definitely important in sensory systems, because the sensory systems have to deal with correlations that come in from the external world that are essentially unavoidable, and it has to essentially combat these types of correlations. For example, here in this image, there's a huge number of pixels are highly correlated and encode for essentially void. You want to get rid of those. So it would be helpful to attenuate the representations of these features. And the retina has obviously found ways to do this for visual stimuli. Olfactory stimuli probably have a very different statistics, and therefore different computations are necessary for this. So what we believe this computation is doing for the olfactory bulb is that it attenuates the representation of functional groups that strongly influence receptor binding and thereby introduce correlations that are maybe not very relevant for an animal. So by suppressing responses that are common responses to these kind of functional groups, it could therefore allow the system to be more sensitive and more specifically represent the informative parts of this. So just a few words at the very end. I've described this computation as a whitening transformation, which is by and large correct, but please understand that this is a whitening of activity across the population of neurons. Whitening has also been discussed in the retina, but it's a whitening across the channels, across the ganglion cell outputs. So this would support efficient coding, which makes sense for the retina because the optical nerve is an information bottleneck. Now, whitening across populations would actually help decorrelate patterns and would help storage in auto-associative memory networks. In fact, in many modeling studies, decorrelation is just performed before you store something in networks. This, I think, is exactly one of the main functions of this transformation in the bulb. And I think there's also an analogy to the hippocampal system where the dentate gyrus decorrelates inputs for CA3 then to store patterns in a associative memory system. And this could be actually to some extent similar in the olfactory system, where the olfactory bulb decorrelates inputs and then piriform cortex can function as an associative memory system. And finally, there have been models derived based on the similarity matching principle that predict circuit architectures for whitening that are actually very similar to these ones here. This is work by Mitya Klosky and colleagues. So with this, just a brief recapitulation. So we found that there's a whitening of odor representation in the olfactory bulb that that is based on specific multisynaptic inhibitory connectivity, and we need wiring diagrams to actually to resolve this kind of connectivity. This depends on tuning, so we also need to combine the reconstruction of wiring diagrams with activity measurements here. And it seems that this connectivity actually represents a memory of molecular features that has presumably been acquired during evolution. We cannot rule out that there's an activity-dependent component of circuit development here. But just because this was in a very young fish, we think it's more innate. Now, this was for us an example to show that there's actually a lot of information in the wiring diagram about the function of the circuit. So by getting this wiring diagram, I would definitely say it was worth the effort. We could understand this. And therefore, this functional connectomics approach may actually be a promising approach also to address other similar questions. And with this, I would like to thank the contributors, and this is first and foremost Adrian Wanner, who has really pioneered this project from A to Z and has done tremendous work that was really pioneering in character. I've also in passing mentioned some work by Thomas Frank and by Peter Ruprecht, and I'm very grateful to Christelle Genoux, our electron microscopy expert, who really gave very important input here. Thank you.
All right, we've got a couple minutes for questions.
Thanks, beautiful talk. I was wondering, how does the connectivity come about? Could Heppian learning, for example, help shaping the connectivity from random?
Just as I mentioned briefly, we think it is probably set up by innate mechanisms, meaning some molecular mechanisms, simply because the system essentially had only 2 days for activity development, dependent development between the time when it was first became odor sensitive to the time point when we actually measured the wiring diagram. I think this is not enough time for any substantial modifications by activity-dependent mechanisms, but we cannot rule this out at this point.
Hi. So how robust is this, the feature suppression system in terms of concentration invariance?
This is something we haven't tested. That's something that we are going to test very soon. For this, of course, simply because we have not varied the concentration of our stimuli. So we can now do exactly this, change the odor set, vary concentrations, etc., and see what's coming out. The good news is that the circuit reconstruction has actually become 10 times faster, and at the same time, the signal-to-noise ratio also increased 10 times. So this now puts us in a position to repeat this experiment in very experimental conditions.
Awesome. Thank you.
I have a question about the timescale of this decorrelation process. From the data slide, it looks like it takes a few hundred milliseconds for the decorrelation process to take effect. What do you think explain this long timescale?
Well, I mentioned this. I think the long timescale is a lot due to the fact that we ramp up our stimulus slowly. And our stimulus, we do this for 2 reasons. One is in order to deliver this to a small zebrafish nose, we have to pass water, which has high viscosity, through thin tubes, and that distorts any odor pulse. And the second is that so all the physiological observations point in the direction that the system is actually not tuned, not made to follow fast changes in odor concentration. These fish live in still waters most of their time. Presumably, there are very shallow gradients in odor that change only slowly when the fish is actually moving through the environment. So we think the system can be faster. The indication we have for this is when we just turn on the input using optogenetics, then the pattern goes to a steady state much faster. So we think the system can be faster. The reason it's so slow is that our stimuli are slow. But I'm not sure whether this is relevant. But in fact, we have not really studied this systematically. It could become interesting to do that.
Thank you.
Okay, I just have one quick question. So you said you did these in very young animals, and the granule cells hadn't come in yet?
Yep.
Is that what you said? So which interneurons are these, do you think, that are doing this process?
So we have, of course, very detailed morphological information, and they look like actually a broad selection of essentially all the upper layer interneurons in the olfactory bulb. This would be periglomerular cells, different subtypes included, and short axon cells. So it seems that these cells are actually enough to do this decorrelation.
To do this really interesting computation.
And the granule cells then come on top of this, and maybe have something to do with actually activity-dependent and experience-dependent modifications of processing that may then be unique to each individual.
Thank you. Well, let's thank Rainer, please. All right, our next speaker is Cindy Poo. She's going to be talking about a map for odors and place in posterior piriform cortex.
Does it work? Yes. Okay. Hi, thank you for the invitation to speak here. I'm very excited to share with you our work looking at the encoding of olfactory and spatial information in the piriform cortex, which we believe is a relatively accessible entry point for a little-known topic, that is the higher order functions within the olfactory system itself. So chemosensation or olfaction is an evolutionarily ancient sense and used by a wide range of organisms from bacteria to the honeybees for spatial behaviors. This, of course, also extends to rodents, which is our model organism, where search behavior using odor plume dynamics as well as odor memory guided navigation are ethologically fundamental. So given the well-characterized properties of navigational circuits in the rodent, we were interested in investigating the interactions of olfactory and spatial information. As Rainer pointed out earlier, in the rodent and rats, the primary cortical representation of odors is in piriform cortex. Here, as opposed to the olfactory bulb, where odor identity is encoded by stereotype maps, projections from the bulb here are transformed such that odors now activated a distributed and random sets of neurons that are no longer topographic. And it is here that neurons are thought to underlie odor recognition and memory. A well-known and interesting property of neurons in piriform cortex is that the circuit is highly recurrent and associative, such that feed-forward drive coming in from the bulb actually only very weakly determine the spiking of piriform neurons. In addition to feed-forward sensory and local associative inputs, the neurons here also receive significant direct projections from the hippocampal formation, and in particular, from both medial and lateral entorhinal cortices, as well as the hippocampus. Now, this suggested to us that spatial and contextual information associated with odors could be actually present in the olfactory system itself. To investigate this possibility, we recorded from the posterior region of piriform cortex in an odor-guided spatial choice task for rats. Okay, so in our task, rats behave on an elevated plus maze that is 1 meter by 1 meter in length. There's a port at the end of each of the arms which can deliver either a light cue, one of 4 possible odors, or a water reward. At the beginning of a trial, a light appears randomly at one of the 4 ports to indicate to the rat that he must approach to initiate the trial by poking in. After poking in, one of 4 possible odors is delivered at random, and the identity of this odor determines the correct reward port for the animals to navigate to and collect water. After a brief inter-trial interval, another trial begins. So just to reiterate, each one of these 4 odors can be sampled at any location, but reward location is dependent on the odor identity only. I will show you an example of one of our rats doing this task. There's going to be 3 trials, and you will see that in this first trial, there's a light at the north port indicating for him to approach and poke in. Let's see. Oops. That did not work. Well, I guess maybe the video is not working. It's playing? It's playing for you guys. Well, not for me. Okay. So you see, he poked in, got odor 3, and went to the east port to get reward. In the next trial, he's going to initiate now at a different port, get the same odor, but the correct choice is still to go east. Okay, and on the subsequent trial, he will sample a different odor at the same location, but now the rule is to go north to collect water. Okay, so as you can see at these example trials, they need to use both olfactory and spatial information to correctly receive a water reward. After some training, rats perform well across odors as well as sampling locations. Now, given the structure, reward contingencies of our task, the most efficient and compact strategy is for rats to use an allocentric map in conjunction with odor identity. However, given the total possible combination of trajectories, it is also possible that instead they're executing a complex egocentric action plan instead, such as when I get odor 1 here, I turn left, etc. How can we distinguish between these two possibilities? Well, given an egocentric strategy where rats keep track of actions that lead to reward, we would expect them to be more likely to repeat recently rewarded actions. Whereas in an allocentric strategies, we would expect them to be more likely to revisit a rewarded location instead. So when we did this analysis, we see that given reward, repeated actions are no more likely than different non-repeated actions, whereas they're more likely to revisit the same rewarded location. In other words, given that a reward was received in the north port on previous trial, rats are more likely to revisit the same location as well as, etc. So we think that in this task, they're likely to be engaging both olfactory as well as the hippocampal entorhinal navigation system. So next, we targeted our recordings to posterior piriform cortex as well as dorsal CA1 in rats using chronically implanted tetrodes. What do neurons look like in piriform cortex? Here I'm showing you one example cell of a neuron in posterior piriform cortex with time aligned to odor onset, and you can see from the PSTH and raster that this cell has an increase in its firing rate in response to this odor. Now, if you look at the firing rate to this odor as a heat map now, where XY axis is the location on the maze, we see that this is a typically odor selective neuron. It is activated by this odor at all 4 of the port locations. And indeed, this was a cell that is purely odor selective, but didn't care about the sampling location. In another cell that's recorded in the same session in the same region, we see that while it is activated by odor 4 as well, has a slightly different temporal profile, which we will get back to in a little bit. But now if we look across space, we can very clearly see that the activity in response to odor is restricted only at the west port for this cell. And looking at all odors, we see that very clearly the cell is indiscriminate for odor identity, but purely spatially selective. And of course, we also see cells that are selective to both odors and locations. For example, this cell prefers odor 1, but only at the north port. Overall, surprisingly, we found that more neurons were location selective than odor selective, and the fraction of jointly selective neurons was slightly higher than predicted by chance. In a pseudopopulation analysis, we found that while both odors and locations can be decoded, fewer numbers of neurons were required to reach 90% accuracy for locations than odors. And in comparison, our CA1 population recorded at the same time reached 90% accuracy with fewer neurons. Odor decoding in the hippocampus was relatively poor in this task, with a total of 91 neurons that we recorded in CA1 performing at the same level as only 5 piriform neurons. So next we asked, what is the temporal relationship between these two representations? To do this, we looked at simultaneously recorded populations. And what you can see here, when we aligned the decoding to odor onset on the x-axis, we surprisingly found that the location classification was above chance and rose even before odors were available. This means that what we have is spatial information that is independent and does not require sensory drive from the bulb. We wondered if the signal that we see is something akin to perhaps a task or context-specific predictive signal, or is it actually a spatial map that is stable and carried throughout different task events? To do this, to address this, we looked at the neural activity in addition to around odor sampling, also at goal and intertrial intervals. And you can very clearly see, particularly in our example cell 2, this very spatially selective cell, that indeed the spatial selectivity is carried throughout different trial epochs. For the population, a classifier that is trained on that activity during initiation can successfully decode spatial locations from neural activity at the goal period, which is on the y-axis here. And even during the intertrial interval, this holds true. Therefore, so here each data point is one simultaneously recorded session. So therefore, what we find is that spatial representations in pure form is actually stable and persistent, and looks like something that is akin to an actual cognitive map for space that is suitable to guide navigation. What is the relationship between these spatial maps here to those of the hippocampus? To begin to look at this, we looked at the coherence of the spike times here to either the sniffing or the hippocampal LFP to understand their functional connectivity to either bottom-up sensory or top-down cognitive maps in the hippocampal network. Here is a coherogram aligned to time from odor onset, and you can see that overall, posterior piriform spikes are coherent to sniffing, with an increase in frequency of coherence at around time 0, reflecting the active sampling of the animal for odors. And the CA1 spikes was coherent to local field potential at the theta band frequency, which characteristically dips around time 0 as animals slow down to sample the odor. While we don't see the same degree of coupling for piriform spikes overall to the hippocampal LFP, which is in the bottom left plot here, we further looked at the coherence based on the decoding performance of individual neurons here for either odors or locations. And when we examine this, we find that the coherence based on the single cell decoding performance for odors showed that they were strongly correlated with the coherence to sniffing and less so to CA1 LFP. And very strikingly, in the opposite direction, for our location decoding neurons, we found that they were selectively coupled to the local field potential of the hippocampus and not to sniffing. So, in summary, what we find is that in rats that use odors to navigate, the spatial information is not only present, but more prominently encoded than odors in posterior piriform cortex. And these maps are stable and persistent across behavioral states, and that potentially a spectrum of different connectivity levels to the bottom-up or the hippocampal networks could mechanistically underlie the spectrum of different odor place selectivity that we observe. So this leaves many open questions and a few intriguing implications to us. First of all, in the context of spatial navigation, this could allow for the anchoring of odor information within a spatial reference frame, allowing for animals to navigate using olfactory landmarks in their environment. And another possibility, which is particularly interesting to me, is that considering the prevalence of map-like representations that have been found throughout the brain, the proposal recently raised by Tim Behrens and colleagues is that these cognitive maps that have been found in a spatial context may actually be an instance of a more general coding mechanism for the brain to organize information of all kinds. Therefore, our observation could also reflect the fact that circuits in posterior piriform cortex are generally suitable for higher level cognitive functions associated with odors. Okay, so to conclude, I would like to, of course, thank Zach for his support in this project, as well as my collaborators Gautam Agarwal and Niccolo Bonnachi, who I worked very closely with on this project, both of them have contributed directly to the results that I presented here today. as well as the rest of the Mainen lab and the funding. Thank you.
All right, we have time for one question. Race.
Do the mixed selective cells show co-tuning to the sniff rhythm and the hippocampal theta?
Yeah, they do. And in fact, we find that it's interesting to look at the co-tuned cells because they, even for the ones that are equally good at encoding locations, they were also coupled to sniffing. Okay. So that we have like the pure odor cell, the pure location cells are not coupled to sniffing, but the ones that are tuned for both are coupled to both.
Great.
Yeah.
Thanks.
Okay, quick. Go ahead, yeah.
Beautiful talk. Have you tried to look at the relation between the auto representations and the spatial representations? For example, I could think you could train your auto decoder on 2 of the arms and test your auto decoder on the 2 other arms and look at differences between CA1 and the peripheral cortex.
Oh. So train only on a subset of the ports?
Yeah.
No, we haven't done that, but that's an interesting idea. Yeah, that would be something good for us to check. Yeah. Okay. Thank you.
Let's thank Cindy.
Thank you.
Okay, our next speaker is Carl Skinover, and he's going to be talking about rapid representational drift in primary olfactory cortex.
The work that I'm going to present today is the fruit of a close collaboration with Andrew Fink. Every aspect of it is as much his as it is mine. Sensory neocortex, by and large, is highly structured, and I hope you'll forgive me for a moment when I gloss over some details and sketch some spherical cows. In the visual system, for example, retinotopy is preserved from the eye to thalamus to cortex by both axon fibers from thalamus and horizontal fibers between cortical neurons, which innervate only specific and local regions of cortex. These synapses, of course, are plastic, and changes in tuning have been observed in cortical neurons over the course of an animal's experience during a task, or as a result of gross perturbation like monocular deprivation. But without these kinds of manipulations, tuning has been found to be grossly stable modulo some noise from day to day for basic features like retinotopy, tonotopy, and somatotopy. Now, individual neurons, of course, can pop in or drop out on a given day, but at a population level, these maps are preserved, and needless to say, this kind of stability is a desirable feature in a sensory area. In fact, it's tempting to speculate that these strong anatomical constraints exist in part to keep a lid on representational drift by setting limits on how far tuning can change, even though these networks are plastic. A few years ago, Andrew and I were struck by some key differences in the circuit organization between sensory neocortex and primary olfactory cortex, or piriform, three-layered paleocortex. Here, input from a glomerulus in the olfactory bulb is distributed across the entire cortex, and the same is true for horizontal connections between pyramidal neurons. And this is what Andrew and I found so striking, that the highly structured anatomy that promotes tuning stability in neocortex is absent in piriform. If there's synaptic plasticity in primary olfactory cortex, it raises a basic question of whether tuning there even can be stable over time. And if representations are continuously drifting, as the anatomy suggests that they should, then what would this teach us about the role of piriform in olfactory perception? Because at first blush, drifting representations might seem like a bug. The experiment that addresses our first question is conceptually very simple and technically very challenging. We just need to measure odor tuning over a long enough interval of time. And to do this, we presented a panel of neutral odorants every 8 days to awake, head-fixed mice while we recorded a fixed population of neurons using a silicon probe chronically implanted in anterior piriform cortex. This is on the opposite end of this cortex from where Cindy was just describing her work. We've spent several years developing the methodology to obtain sufficiently stable recordings, and I'm going to show you one representative example of this. These are the waveforms of a single unit as they register on the 32 sites of our probe on day 0 of an experiment. And now superimposed is this same single unit recorded 1 day later, and superimposed again every day for the next 16 days. In addition to this example, we have a large body of evidence that demonstrates our ability to follow the same neurons over time. Please come find us afterwards if you're an aficionado, or if you would just like to kick the tires. Some single units show stable tuning. These are responses to a panel of 6 odors on day 0, and they're virtually unchanged 16 days later. But some units are less well behaved. This one shows strong modulation to odorants 1 and 6 on day 0, which is mostly lost and replaced by a vigorous response to odor 3. And this second example is the rule, not the exception. This shows a random subset of 300 odor unit pairs. Each row is the z-scored response of 1 unit to 1 odor, sorted according to response magnitude on day 0. 8 days later, a lot of this structure is preserved if we maintain the same sorting as on day 0, but over longer time periods, it gradually falls apart. Now, it may seem like it's not so much that responses are reorganizing, but rather that they're diminishing. But if we sort responses within each individual day, we can see that this is not the case, and indeed, roughly the same number of neurons respond to odor on every day. It's just that this response is profoundly reorganizing over time. It also appears that these changes are cumulative. To test this, we constructed a stationarity index which quantifies drift across days while normalizing differences between stimuli and animals. It's simply the differences between within and across day correlations of single trial population vectors and evaluates to 0 when responses are stable across a pair of days with negative values indicating greater drift. Here are the cumulative distributions of stationarity indices for intervals of 8, 16, 24, and 32 days, indicating progressively increasing drift. In fact, by the end, that drift is so pronounced that they're just grazing the distribution with shuffled stimulus labels. So changes in population tuning accumulate over time. But what if the most informative dimensions in the population response about stimulus identity nonetheless remain stable over the long term? To examine this, we trained a linear classifier on day 0 and tested it on later days. Cross-validated performance on day 0 is of course high, but classification accuracy steadily degrades over time, approaching chance after 32 days. So odor responses in piriform, which lies just 2 synapses downstream of the nose, exhibit gradual, cumulative, and profound reorganization over time. What might this imply for the role of piriform in odor identification? It certainly doesn't seem like it's a good place to read out stimulus identity, at least over the long term. That doesn't mean that it plays no role in perception, only that it might not be a reliable source of ground truth information about what's coming in at the nose. Now, we could imagine mechanisms by which a downstream reader could compensate for this gradual drift by, for example, leveraging some invariant geometry in the population representation. Alternatively, if you need reliable information about what's coming in at the nose, just ask the olfactory bulb. In addition to piriform, the bulb forms direct projections to several key regions that link sensation and action, and it's been shown that in the absence of a task, tuning in bulb is constant over months. So other regions don't necessarily even need the piriform to find out about what's coming in at the nose, at least over the long term. Meanwhile, piriform is taking perfectly stable input from the bulb and scrambling it over time. So what is piriform good for? There's an old idea that's been mentioned a few times this morning already that piriform cortex is a dedicated learning system for olfaction. This has received renewed attention in recent years, including by the 2 previous speakers in this session. Now, what exactly it is that piriform might be learning and how remains a matter of debate. In fact, Cindy Poo and I have organized a workshop on Tuesday to examine this very question. But regardless, if piriform is operating as a learning system, we wondered whether the representational drift that we've uncovered might reflect an ongoing learning process, and we've made 2 observations that support this view. First, we found that frequent experience with a stimulus stabilizes its representation. We presented a set of odorants every day for 16 days. Then, we continued presenting the now familiar odors daily, but also presented a new set of odors only every 8 days. And the representations of the familiar odors then barely moved, both across the 8 and the 16-day intervals. In contrast, the representations of the novel odorants drifted much faster. So daily experience stabilizes odor representations. And the fact that we can observe both stable and drifting representations in the same tissue, recorded using the same probe, spike sorting using the same pipeline, argues that the changes in odor tuning that we're observing are not just because of a failure to follow the same neurons. What if the same process that causes stabilization when odors are encountered daily is also what causes drift when odors are encountered only infrequently? These two phenomena might seem like they're at odds with one another, but there's a parsimonious account of them under which they're just two sides of the same coin, and that is if what we're dealing with here is a fast learning system that is continuously overwriting itself. Stefano Fussi and colleagues characterized a fundamental tension of learning system as follows. The main challenge in building models of long-lasting memory is protecting the memory trace from the ravages of ongoing activity. Without a compensatory mechanism like Fussi's cascade model, a learning system overwrites itself at a rate proportional to the rate at which it learns. Could drift in pure form be a manifestation of this kind of overwriting? It's not as if our mice don't sniff during the 7 days in between each measurement. They're constantly encountering other molecules, like the ones in their home cage or on the husbandry technicians or on us. Perhaps then, drift in pure form is simply a consequence of a continual learning and a continual overwriting. This thinking motivated a third and final experiment in which we asked whether memory traces formed through experience can be stored, or whether pure form is a fast learning system in which memory traces are quickly overwritten due to the ravages of ongoing activity. Once again, we stabilize odor representations through regular experience, but importantly, we then halt daily presentation. And this reverses the effects. The representation of familiar odors begins to drift again, so quickly, in fact, that 16 days out, they're moving as fast as the novels are. So memory traces formed in this system are not burnt in. We've made 3 observations. Stimulus representations in pure form drift, exhibiting rapid and cumulative reorganization, even in the absence of a task, simply as a function of time. Daily exposure stabilizes these representations, but without that ongoing experience, they're not stored over the long term and begin to drift once again. In our current working model of the olfactory system, the bulb, and not the pure form, conveys ground truth information to the rest of the brain about what's coming in at the nose. Pure form operates in parallel as a fast learning system, an extremely flexible, short-term scratch pad that can learn the structure of the odor environment with only limited input. And this is reminiscent of an old hypothesis that pure form is a kind of hippocampus for odor, capable of picking out any structure in the olfactory environment that's relevant to the animal. So for instance, Cindy Poo's just presented evidence that when an animal is performing a spatial task, posterior pure form encodes location. Kevin Franks has argued that it implements pattern completion even after limited experience. Andrew and I had a poster on Thursday with preliminary data indicating that pure form can learn both the identities and the temporal relationships of elements in an odor sequence. What is clear, though, is that it doesn't seem to hang on to what it's learned over the long term. And if pure form can't bake in memory traces that it encodes, this again raises the question of what it's good for. The hippocampus literature has been grappling with this exact same problem for a long time, which has led to the theory of consolidation. Extending this hypothesis to the olfactory system predicts that there's a slow learning system downstream of both pure form and bulb. And under this view, these 3 systems would work in concert, leveraging ground truth information about what's coming in at the nose conveyed by bulb, fast learning in pure form, and long-term storage in a hypothetical slow learning system. And this is a possibility that we are actively investigating. Thank you.
All right, we got time for a couple questions.
Carl, do you think that replay in piriform cortex is stabilizing preferentially the novel odors?
Do you mean locally or to teach it to a downstream region?
Your choice.
We are keen on the second option, although we do not have good evidence for it yet. But I mentioned, one way you could recover information is by sort of knowing something about the geometry of the representations and sort of keeping up with that drift slowly over time. Another would be to exploit something like replay to sort of continuously teach everybody downstream what's going on. Yeah.
Can I ask for 2 speculations? One is what are candidates for the downstream area? And the second is if you pair an odor with a very strong valence, do you think there are mechanisms that could stabilize it for longer?
So I can speculate about the first question and answer the second. First of all, we don't know. We're actively speculating ourselves. There's one region downstream of the olfactory bulb that doesn't obey the circuitry logic that I presented at the beginning. It actually receives patterned input from the bulb, and for all we know, local connectivity also is patterned, and that's the cortical amygdala, which sits basically towards the back of the piriform, right nearby actually Cindy Poo's neighborhood. So that's an extremely attractive candidate because it receives both direct input from bulb that's patterned and also direct input from the piriform cortex. So that is one of our preferred hypotheses. To answer your second question, we did the following experiment. We wondered whether if we afforded salience to a molecule, whether the representation of that molecule would be more stable than the representation of neutral molecules. So to do that, we simply paired a molecule with shock and taught an animal to fear it. And when you do that, you can then wait 16 days, well, measure representations, then wait 16 days again, and look at how much the representation of the CS plus has drifted relative to other neutral odors. So here are the stationarity indices for the neutrals, and here are the stationarity indices for the conditioned. So initially, when we did this experiment, we were both disappointed and surprised and couldn't make sense of it. In light of what we know now, actually, there's a very simple explanation for it, which is this cortex doesn't seem to be learning necessarily about salience. And in fact, we only presented the CS plus 8 times in this animal's life, 2 weeks prior to the final recording. And if what this region is learning about has more to do with, say, the statistics of the odor environment, regularities in it, then those 8 stimuli probably barely registered. So there's a sort of satisfying explanation, I think, here, because we have this extremely salient stimulus that the animal knows exactly what to do when it encounters it, and yet the piriform doesn't seem to have stabilized it, because we think this indicates that that's not the kind of stuff that the piriform is learning about.
So actually, this is related to the answer that you gave to the first question. So have you looked at the geometry in the latent space and whether it's invariant over time or not?
Did you say in the valence space?
No, in the latent space.
Yeah. Yes, the geometry is quite stable over time. It's not entirely surprising, given what we know, or we don't know a ton, but the nonlinearity that's applied in the transformation from bulb to cortex is actually pretty soft, which means that a lot of correlations that exist in bulb basically get imported in cortex. What correlations remain after Rainer has sort of scrambled it up, I should say. Actually, a lot of the structure that you would see in bulb, you're going to find in cortex in some sort of somewhat diminished form, but nonetheless still there. And so something that sort of falls out of that by default is that any geometry that you would encounter in the bulb, you will see in the piriform. And so no matter how the piriform is drifting necessarily, that sort of overall geometry at a population level is roughly constant. And Bob Dada and his graduate student Stan, sorry, I'm going to butcher his name, so I'm not even going to try, have some very beautiful data to this effect, looking specifically at molecules that are similar to one another, both in percept and also in their structure. And they've demonstrated convincingly that there is preservation of local geometry.
Right, so that solves the problem in a way then, right? You don't need downstream consolidation.
No, it solves it only if a region downstream can keep up with it. So now we need to posit a mechanism to sort of keep up with those rotations in this representation space, which is entirely possible. There's no reason why that wouldn't exist. What we're proposing is there's actually a much simpler solution, which is given that the piriform is breaking the china, ignore the piriform and just listen to the thing that didn't break it, namely the bulb, since it's sending out parallel projections. But it's entirely possible that regions downstream can also keep up with drift, and I will use this opportunity to plug another workshop on representational drift, in which these kinds of models are going to be discussed. Actually, that's tomorrow, Monday.
Min Chung, and he will be talking about manipulating and synthetic optogenetic odors reveals about the coding of olfactory perception.
Sorry for the overly long title. So thank you everyone for coming today. Today I'll be talking about my work in the Rinberg lab, combining optogenetics and behavior to understand the olfactory code. So as we've already seen from Rainer's slides, we know that different chemical odorants evoke patterns of activity in the olfactory bulb. These patterns are unique in space, which glomeruli are activated, and they're also unique in time, when they're activated. So my research asks the question of what features within these activity patterns are causal for perception. For example, if I were to change a pattern in space or time, would the animal still smell the same odor? To address these questions, we turn to optogenetics, which allows us the ability to directly manipulate these patterns in space and time in a precise and parametric manner. We use a mouse line in which channel rhodopsin is expressed in all olfactory glomeruli. We implant a chronic cranial window over olfactory bulb, giving us long-term access for optogenetic stimulation. On the olfactory bulb, we then project these light patterns. Here, each square spot is 120 microns, comparable to single glomerular size. And because glomeruli tile the surface of the olfactory bulb, essentially my light patterns evoke unique spatial-temporal combinations of glomeruli. Without going into technical detail, my stimulation system allows me to stimulate at a resolution of single millisecond and 10 micrometers. We then proceed to train animals to recognize these optogenetic patterns or synthetic odors. We use a two-choice task in which animals either lick left or right for water rewards. I first define a set of target patterns where animals are trained to lick left, for example. Over the olfactory bulb, I randomly initialize 6 spots according to some timing, defined relative to the sniffing or the respiration of the animal, where 0 marks inhalation onset. These 6 spots are activated in a strict temporal sequence, where spot 1 is activated first and spot 6 last, with 80 milliseconds durations of spots and 40 milliseconds lag between spots. I then define a second set of patterns, non-target patterns, which are 6 other off-target spots, randomly chosen from trial to trial, with timing also randomly chosen from trial to trial within a comparable time window of a single sniff. Critically, after mice are well trained to do this task, I present on a small number of trials, probe trials, which are perturbations of target patterns. These perturbations can be spatial, which are replacing one or multiple spots in the target pattern, or they can be temporal, which are shifting in time, one or multiple spots in the target pattern. We can conceptualize the responses of animals as the following. So if I train the animal to lick left to the target pattern, and for a well-trained animal, if I present the target patterns 100 times, the animal is going to lick left 90 out of 100 times, the point in red. And if I present a non-target pattern 100 times, the animal licks left 10 out of 100 times, or the point in blue, establishing a non-target baseline response. Critically, we want to see what happens on probe trials along some perturbation dimension. For example, let's say timing of the spots is completely irrelevant to the animal in performing the task. If so, no matter how much I change the timing of the spots, the animal is still always going to lick towards the left. In contrast, if timing is extremely important, for small perturbations in timing, the animal is going to lick towards the right, with the slope of these responses reflecting the importance of each feature dimension to the animal's recognition of the different patterns. Let us first look at the results for replacing one or multiple spots in a pattern. First, I plot the effect of increasing the number of spots replaced in a pattern, and as before, I plot the baseline target in blue and non-target in the dashed line responses. So here you see as I increase the number of spots that are replaced in a pattern from 1 to 5, the animal licks to the left less and less in a graded, almost linear fashion. Now there are 6 different ways to replace one spot in a pattern. There are 15 different ways to replace 2 spots in a pattern, and so on and so forth. And here I plot the animal's responses to every unique combination of spots replaced. let's first look at the results for one spot replacement plotted by spot position, where spot 1 is the first spot and spot 6 is the last spot in the pattern. Here, you can see that there is a stronger effect of replacing the earlier compared to later spots in the pattern. We refer to this as a primacy effect, consistent with what previous studies have found. Now, we want to try and predict the responses of the animals to every single combination of spots replaced. And it turns out, despite the apparent complexity in the data, the simplest possible model can explain the results, the simplest model being a linear readout. For example, if the effect of replacing one spot in the pattern is X, and the effect of replacing another spot in the pattern is Y, then the effect of replacing both spots at the same time is some function of X plus Y. Hence, at least for spatial perturbations, individual spot effects are additive, meaning the effect of replacing any individual spot is independent of any other spot being replaced. Now we can look at the data for shifting in time spots in the pattern. To keep things simple, as before, we can examine the results for single spot shifts while keeping the timing of all other spots in the pattern constant. As before, I plot these shifts by spot position, where spot 1 is the first spot and spot 6 is the last spot in the pattern. Spots can be shifted in two directions, in the positive direction or later in the sniff cycle. And here are the responses. Spots can also be shifted in the negative direction or earlier in the sniff cycle, and here are the responses. So there are two major observations from this data. The first that you can see is that there's a larger effect of shifting the earlier compared to later spots in the pattern, consistent with the previously found primacy effect. And the second is that you can see an asymmetry in the responses. For example, if you look at spot 5, there's a stronger effect of shifting the spot earlier compared to later. And we like to think of this asymmetry effect as being consistent with the primacy hypothesis, because while the later activated spots in the pattern may be less important, as you start shifting them earlier and earlier, they start to interfere with the early, more important representations. Now we can proceed to model the effects of each individual spot shifted in either the negative or positive direction. And then we can use this simple model to try and predict the animal's responses to all temporal perturbations. However, we ran into some trouble with one additional experiment. So in this experiment, we took the entire target pattern and shifted it all together at the same time with respect to inhalation. Under this manipulation, we see only a weak effect on behavior responses. And critically, if we take the data on the previous slide, where we know the effect of shifting each spot individually, and we sum up over all spots in the pattern, we see that the summed prediction greatly overestimates the effect on behavior. So what's going on here? What's going on seems to be that when you take the entire target pattern and shift it all together at the same time, what you're changing is the latency of each spot relative to inhalation, but you're completely preserving the relative timing between spots. Hence, it seems to be that animals seem to strongly encode relative timing between spots, and we further verified this with simple regression models showing that models based on relative timing outperform models based on inhalation-locked timing. So now we want to put everything together into an overall process model which explains how animals go from spatial temporal glomerular inputs to perceptual outputs. And we have seen several things that seem to work so far. So the first is we've seen a linear sum of spatial effects. We've seen a primacy effect, where earlier activated spots are more important. And lastly, we've seen an effect of relative timing. So we propose the following model based on template matching. Under this model, animals have to discriminate between some learned target pattern versus some new incoming probe pattern. Patterns are defined as activations in discrete spot, or more generally, glomerular channels. Then we perform very simple transformations on these patterns such that they take on exponentially decaying waveforms. But critically, we model the height of these waveforms such that the earlier activated waveforms take on larger amplitudes than the later activated waveforms. And this captures the primacy effect. We apply this transformation across all patterns. And next, we perform a simple template matching procedure to compare the two sets of patterns. And this comparison is done within each spot channel by comparing the differences between waveforms. However, because waveforms are currently defined with respect to inhalation onset, then what this comparison reflects are essentially differences in inhalation locked timing, which is not what we want. What we want for the model to reflect is relative timing between spots. So to do that, for each set of patterns, we further define a quantity of center of mass, which captures the overall position of each pattern within the sniff cycle. Hence, now individual waveforms are referenced to its own pattern, center of mass, as opposed to the inhalation onset. And now when we perform the comparison of waveforms aligned to the same center of mass, differences between waveforms essentially reflect changes in relative timing in spots. And finally, we take all these waveform differences, sum them up linearly across channels to produce a single quantity or perceptual distance, which we then use to predict the responses of the mice. So, the larger the perceptual distance, the larger the predicted perceived difference between the probe pattern and the target pattern. And here, I'll plot the predictions of the model against the data. Points which fall on the unity line indicate perfect prediction, and deviations indicate poor predictions. For reference, here are model predictions on shuffled data labels. And finally, here are model predictions across all spatial or temporal perturbations that we perform, of which you've seen a subset today. Hence, a simple model containing just a few critical components manages to capture the essential computations that the olfactory system performs to generate perception. So to summarize, we train animals to recognize synthetic odor patterns. We perform precise and parametric perturbations on these synthetic patterns while measuring changes in perception. And this allowed us to derive a single computational model which explains how animals go from spatial temporal glomerular inputs to perceptual outputs. And I'd like to thank my various collaborators as pictured above, my PI Dima Rinberg, and in particular my collaborators in Italy, Stefano Panseri and Monica Moroni, with whom I worked to develop the various mathematical models. I'd also like to thank the larger NYU community, including my thesis committee, Kathy, Tony, and Weiji, who provided invaluable suggestions throughout the project. Thank you very much.
All right, we have time for one more question.
Hi, if I understand correctly, it seems like when you did your temporal manipulation, you also affected the correlations by moving one odor a little bit sooner or later. You induced some additional synchrony in the signal.
Mm-hmm.
I was wondering if you thought about just randomizing the order of the odors, or not the odors, but the stimulation while maintaining the same temporal pattern.
Yeah, so basically, we did that manipulation where we just scrambled the order without changing the fine timing. And we find that that produces some decrement in the behavior. And then on top of that, if you additionally perturb the fine timing in addition to just the order, you get an additional decrement in behavior on top of that. So it's not just the sequence that matters, it's actually the fine timing of each spot relative to other spots. So you can see this more clearly if I go back a few slides to the single spot shift data. Let me bring that up quickly. So if you look here, for example, So if you look here, for example, if you look at a single spot shift, so each dot there is an increment of 20 milliseconds. So those small increments basically still preserve the order between spots, and yet you still see changes in behavior under those manipulations.
But what if you permute the order? If you just permute the order?
Yes, so if you permute the order, you see changes in behavior.
Okay, because that should completely change the relative timing between the
Yes, that's part of it. That's correct. Yeah, thanks.
All right, let's thank all of our speakers for this session. And we will reconvene for our final session at 2 p.m.